overseas professors who are coming on this platform and will deliver the lectures with their expertise. And all of us know, all of us are eagerly expecting or expectantly waiting for their deliberations. Uh, Before I say a few words on vector-borne diseases, let me tell you for the overseas professors that the West Bengal State University is a very young university, very a toddler basically, just 12 years old. And West Bengal State University, right from the beginning, uh, started to use information technology and biotechnology and other technologies to conduct the operations of the university. Today, when the pandemic is causing a lot of problems for different institutions, the West Bengal State University, because of the wisdom that it had shown in the year 2008 of doing business through internet, is still doing business through internet without much of a problem. I do not know, and I am not comparing with other universities because that is not proper, that is premature. Every university has its own character, every university has its own strength, every university has its own weakness. In spite of all the strength and weakness, one thing can be said: one after the other, the quality of the webinar that is happening, one after the other, and the quality of the speakers and the quality of the discussion that is happening simply overwhelms all of us. And the academic content of these webinars is very strong, very solid. And with that, the departments are trying to make people aware of different subjects. Some webinars are on pandemic and how to do the teaching learning process. Some webinar is on different platforms, how those platforms can be utilized. Some webinars are on uh, uh, some webinars are on uh, uh, subjects like physics, where the alumni are coming to speak on their frontiers of research. And today we have the overseas visitors, and I think this is the first webinar where all overseas professors are coming on the platform to speak on their expertise. Now, vector-borne diseases, which is the subject matter of today's webinar, that account for more than 17% of the infectious diseases, causing lakhs of death annually. I do not know exactly the figure of death, but causing a large number of deaths annually. They can be caused by either parasites, bacteria, and various other pathogens. Targeting the vector host interface by incorporating vector salivary proteins in antipathogens vaccines is a recent approach along with the second generation recombinant pathogens. The debate remains as to whether vector saliva exposure protects or reduces the protection against more severe clinical manifestations, induces immunity through natural exposure, or extends to all vector species or associated pathogens. The subject is of great interest because it directly relates to the protection of human health and human lives. Exploiting the unique biology holds promise for all of us. It is a great strategy, or it may be a great strategy for the development of vaccines against vector transmitted diseases or different other viruses. All of us know the background of this, or the perspective of this whole discussion, that today this SARS-CoV-2 virus that is causing havoc basically all over the world. And after even in the Second World War, not so many countries got affected the way SARS-CoV-2 has caused problems. Already large number of people have been affected. A lot of deaths have taken place, and On top of that, this lockdown has confined us to our houses. We cannot go out. There are many restrictions which are put on people who are active in their professions, children, and other people of different ages who go to colleges and universities, and so many other people. So it is indeed a very great what I should say, calamity that has come upon us. So it is necessary to understand, and today the vaccine research will be discussed to some extent. Many of us who are ordinary people, laymen, we are thinking why the vaccine is not coming immediately after the virus has spread its wing, spread its lethal activities. But the point is that development of vaccine is not an easy job, and I think the respected professors who have come over here will be able to tell us why and how the vaccines are important and how they are developed. Three very distinguished scientists and professors are with us today. Professor Hira Nakashi, a very well-known Leishmania vaccine developer, and second generation Leishmania vaccine, or third generation, I do not know exactly because I am not a biologist. Professor Pal, Professor Ghorai, and others, they are the people who will know. Professor Chiranjib and others will be able to tell you. Professor Shaden Kamhawai, and Professor Jesus G. Valenjula, they are also there today. They are very active in research on vaccine development. These the two last scientists, Professor Shadain and Professor Jesus, they are the key scientists involved in the recent vaccine trial of the universal mosquito vaccine in humans by the National Institute of Health of the United States of America. All of you know it is a great institute for vaccine development and different other diseases. There is also CDC, Center for Disease Control, or things, or organizations like that. So they are developing vaccines for different diseases. So it is a great day for the West Bengal State University. The IQAC must be lauded for the help that it is giving very silently. The IQAC does not propagate its name. It is keeping the department at the front, and that is the job of this kind of cell. That it will just generate interest among people, and that's what is being done under the leadership of Professor Hota. The IQS is doing its job very nicely. So I am very happy today. Whenever something good happens to West Bengal State University, it becomes celebration for me. What more can I do? The teachers are the real strength of the university. The teachers are the real strength of the university. The researchers are the real strength of the university. And I particularly I take this opportunity to particularly congratulate Professor Chiranjib Pal for leading this webinar and for organizing everything. I think all of you, all the teachers, one after the other, will continue doing this activity and bringing great people on this platform. I say great people because scientists are people who are needed today at this hour of crisis. We only people on whom we can fall back. We can fall back on God, who is not seen, and we can fall back on scientists who are seen and who are seen doing work actively. So I'm indeed grateful to them who have come from miles away to discuss on vaccine development. I am indeed pleased with my teachers because they are my real strength. They are the people whom I love, adore, respect, and congratulate. So they are the persons. They will They are organizing the program and they will lead the program. I was to welcome all of you, as has been stated by Dr. Chiranjib Pal, Professor Chiranjib Pal, and I welcome all of you, particularly those three overseas professors who are our guests today and who will enlighten us with their wisdom and with their methodology of working. That is what is needed for our students, and I'm sure the students and researcher will listen to them and will get a lot of information and foresight as to how research should be conducted. Thank you very much. Stay safe, take care, and follow all the norms that should be given and that should be followed at this COVID hour. Thank you. Thank you very much.
Thank you, sir. Thank you very much for your welcome inaugural address. I would just like to request Professor Orun Hota to say something as a welcome address before to start Professor Hiranakar. Please, Professor Orun Hota.
Okay. Good evening to all of you, and a warm welcome to this overseas webinar on vaccines from vectors for vector-borne diseases, organized by the Department of Zoology and in collaboration with IQAC West Bengal State University. Uh, it is always a great pleasure to listen to our Honorable Vice Chancellor, who is a very good speaker and always inspire us and gives us directions for the development of the university in academically. Uh, I I must acknowledge, or rather, I would like to put on record that under his able leadership, we are progressing in a good path, and the university is developing. And I hope in future also we would not be we would be privileged uh, to get so. Thank you, sir, for your inspiring speech and for all your because our honorable vice chancellor has never interfered in any academic matter so far, and this promotes us, this enlightens us how to go for the development of the university. Really, I am grateful. The university is grateful, and under his visionary ideas and new thoughts and insights, the university will get a name uh, and fame in future also. I would also like to put on record that the Department of Geology has been constantly engaged in research and teaching activities since the inception of our university. Whenever I visit to the department, I find they are they all are engaged in either in research or in other works when they have no classes. It is not like other departments where some of the departments they pass time in gossiping. But it is only department where I see all of them are very busy and they are good researchers, by which they have earned many grants from the government of West Bengal, from the different granting agencies, and from government of India also. So it is a good sign, and it is a good sign that our university, uh, I mean the Department of Geology, is showing some path to other departments also. In this context, I must also put on record that my departmental colleagues are not only engaged in research and teaching, but they are also equally participating in all the committee, actively participating in all the committee meetings and contributing their best for the development of the university. This is a great sign for a department. In this context, I welcome Professor Chiranjib Bhattacharya, Professor Narayan Ghodai, which is a good friend of mine since the last 30 years. 3 decades have passed, and Professor Chiranjib Paul, Dr. Samir Kumar Saha, Dr. Dev Prasad Mandal, and Dr. Somi Bhattacharya. I am thankful to the department for contributing for the betterment of the university. As our Honorable Vice Chancellor told, ours is a very small university and it is only of 12 years. And during these 12 years, we are trying our best in spite of many obstacles, many difficulties. We are trying for the good work and for the betterment of the university. As our Honorable Vice Chancellor sir said, today is a historical day in the context that we have three great scientists, Professor Hiranakashi, Director Division of Emerging and Transfusion Center for Biologic Evaluation and Research, US Food and Drug Administration, Silver Spring, United States. We have also Professor Saden Kamhai, who is a core scientist, vector molecular biology section. He is from Rockville, MD, USA, and also an illuminating scholar, Professor Jesus G. Venenzuela, Chief Vector Molecular Biology, uh, who have uh, who have accepted our invitation, and well, I hope we will be very much enriched, enlightened by their talks, and we will know many things regarding these vector-borne diseases. I mean the vaccines from the vectors for vector-borne diseases. It is my duty to welcome to all the overseas scholars, scientists, and I also welcome my esteemed colleagues of the department, teachers, teachers of our affiliated colleges. I must tell that without their help, the university cannot grow, because and we are very much fortunate that the teachers of our affiliated colleges have been doing a tremendous job by for the betterment of the university under the guidance of PG Department of Zoology. So I welcome to the researchers, participants, and my dear students of the university, and awaiting for an illuminating and informative, what I say, webinar, overseas webinar. And it is a great day for us. It is a great day for us, for IQAC and for the West Bengal State University to have great three scientists and researchers who will be who will enrich us by their talks, awaiting a grand success of this semi webinar. Thank you, Professor Paul. Over to Professor Paul.
Thank you, sir. Thank you, Professor Arun. Uh, Sir, Professor Hiral Nakashi, can we start? Hello, sir. Please.
Can you hear me? Can you
Yeah.
hear me?
Yes, sir. Yes, sir.
Yeah. Yes, we can. If you, whenever you want me to do that.
Okay. So.
Right now.
Yeah, sure. Just a brief introduction.
Okay.
Now, I would like to introduce Dr. Hira El Nakashi. He is the director, Division of Emerging and Transmitted Diseases, Center for Biologics Evaluation and Research, US Food and Drug Administration, Silver Spring, United States. He is the pioneer in the field of vaccine development by gene knockout attenuated parasites against leishmaniasis. He has published more than 130 esteemed publications in the field of infectious diseases. He is one of the key principal investigators in developing a second generation vaccine with a markerless attenuated leishmania major strain using CRISPR gene editing. Today, he will share the experiences with his talk on way to leishmania vaccine development, how second generation leishmanization using genetically modified parasites came of age. Please, Professor Nakash.
Thank you. Let me see if I can handle this technology here. So,
sir, right lower corner, present now.
Oh, that's what I'm looking at it. So I got it. So I will say, show your entire screen. Okay, let's see. Share. Can you see?
Yeah, perfectly. No. Perfect.
Okay, all right. So good morning to my colleagues in US and good afternoon, my colleagues in India. First of all, it's an honor to be being invited to the University of West Bengal University, and I appreciate Chiranjib's request to give a talk. And Chiranjib and I know for a little while he was working in Dr. Jesus's Valenzuela's lab. So I think, first of all, thank you so much. And what I want to do today is to tell you a story about how we came up to this second generation leishmanization using genetically modified parasites. And so where we think we are going and where we are at this time. So this story began sometime 20 years ago when I started dabbling with the Leishmania parasites. So, alrighty, so now how do I move to this next thing? Okay. Oh, God.
Sir,
Now,
make the
so
slide,
how do I move?
Sir, make the slide zoom at your laptop.
Do what?
Zoom. Just make the slides zoom presentation.
Slideshow.
Slideshow.
Slideshow. You are presenting. Where do I see that slideshow?
Lower right corner.
Lower right corner. I don't meeting detail. I don't see. Oh. You mean sorry about that. 1 second. So
Sir,
I'm looking at.
Sir, on your PowerPoint presentation, please go to your PowerPoint presentation.
Yeah.
Yeah.
That's the problem. I cannot see. Oh, okay. All right. So, okay. 1 second. Okay. All right.
Yeah.
So now I got.
That's
All right. So let me give you a little background about. Sorry about the disruption. So give you background about the parasite Leishmania. So Leishmania parasite is it has a digenic life cycle starting from it resides into the vector, which is the sandfly vector, and you will hear quite a bit today by the other two speakers, my other two colleagues, about the sandfly and the saliva and other things, and what it is an important aspect of the parasite development. And so, and the other part of it is that in the vertebrate host. So when the parasite mature, is infected is in the sandfly, it matures into different forms. And then when the sandfly has to take a blood meal from a vertebrate host, in this case happens to be human being, and it injects the parasite into the host and sucks the blood from the host. So when it sucks, the form which is present in the sandfly vector is called, there are different forms, but then overall they are called the promastogode form. These are elliptical, long, motile with a long flagellum. But then there are also different forms, which is in the slide shows the different forms of that. When they are taken up, when they bite, the sandfly bites the human vertebrate host, in this case human, these are taken up by the promastogodes are taken up by the phagocytic cells such as macrophages and other types of mononuclear phagocytic cells. They transform into another form called the amastogode form, which is more virulent form, and then they multiply. Then infect other macrophages. Then the sandfly comes back and the sandfly comes back, takes a blood meal, and then basically the cycle completes. So there is two forms. The one is the sandfly form, and then the vertebrate host. So when we are talking about a vaccine, we are talking about a vaccine which will protect against that naturally infected natural infection. So next slide, please. Okay, next slide. So there are several forms of Leishmania. Many of you know, and I'm sorry if I'm boring others. So there are three forms: the visceral leishmaniasis, the cutaneous, and the mucocutaneous. Visceral is basically the causes fever, weight loss, hepatosplenomegaly, anemia, and if it is not treated, it is very fatal. It's just people die. However, even in certain cases, these visceral leishmaniasis, after treatment, especially in Sudan region as well as in India, then they recover. Then some of them develop this post-Kala-azar dermal leishmaniasis, which is different forms, and they're macular and nodular form. And it is thought that this may be the form which may be important for maintaining the transmission of the disease as a reservoir in that area. And the other forms are cutaneous leishmaniasis. You can see a lesion formation, and but the good thing about this is self-healing and non-fatal. But the other form, which is the mucocutaneous, which causes a lot of destruction of the tissue, and it's very bad. Now, so the global distribution. So why are we talking about, as esteemed Vice Chancellor said, they're infectious disease, and there are a lot of there's global nature to them. So there are about leishmaniasis, about 1.2 billion people are at risk, 12 million are infected. Every year there are almost 1 million cases, and there are more than 50,000 deaths. And then also the interesting part is that now this leishmaniasis comes up in areas where it was not before, in non-endemic areas, and people with a compromised immune system, such as HIV co-infection. Then, especially this disease is among the poverty, people who are malnourished and poor, and then when in the world, there are a lot of disturbances going on, and because and the environmental changes such as deforestation, building of dams, irrigation schemes, urbanization, and things like that. So you can see that in both at the cutaneous form up on the panel A and the visceral form, it is present in many, many countries and all over the world, mostly in tropical and subtropical region. Next. Okay. Next, so I think now why are we interested in the looking at our in the U.S.? Obviously, we are interested in the global infectious diseases, but also it is not that we are completely spared of the disease. There are cases. First of all, there are dogs which have some infection in the U.S. Then there are autochthonous cases recently reported in the southern part of United States because bordering Mexico. So there are cases here, and those people who have never left the country and have been shown that they have the disease. Now, specifically zeroing on to India, it is almost 10% of the people. Country profile shows that 10% of people are at risk. And interestingly, that even though WHO has claimed and that we should be eliminating by 2020, we are already in 2020, and the elimination program country says about 1 in 10,000. If we get that case is 1 in 10,000, and then we are under elimination program is achieved. However, that has not been the case. Now WHO is thinking that by 2030 we should be able to achieve that using and identifying the cases, indoor spraying, treatment of the people as soon as we see that, as soon as the cases are identified. However, as we know, even though treatment and indoor sparing and everything, this disease comes in a cyclic form every 10, 15 years, because what happens is once the caseload goes down, people, slack, and the infection goes up. Unless and same thing, not only cutaneous form disease. Now we have a what is it called, the visceral disease. We have also the cutaneous disease coming up in certain areas, and there's many of you may know that there are Leishmania danavani, which causes now cutaneous disease in certain parts of India, like in the upper, Himachal Pradesh and other areas. And recently there were cases found in the Kerala state, which, these are Danavani cases, and they were had cutaneous, such as in Sri Lanka, people have shown. And recently people have shown that these Danavani has become a hybrid with L major and other tropical genes in there. So it has converted into a cutaneous form. Now, this is the area basically showing, concentrated area where the disease is. And unfortunately, this is in your area where, West Bengal, such as in Sri Lanka, people have shown. And recently, people have shown that these and other areas, which has many cases, and these, just to show you that, even though they are showing the incidence is going in certain areas, but the incidence comes up in other areas. So it is there, and there are several, treatments for this leishmaniasis, drug treatments, starting from the antimony treatment, amphotericin, ambisone, multiphenacin, permamycin. All these treatments are lengthy. You need, you need to go, in many cases, hospitalized. And what happens is that when people take these medicines or are treated for a little while, they feel better, and then they just stop treatment. And what happens is, therefore, the lack of compliance in treatment results in the parasite resistance to these drugs. And every drug which has been tested has now developed resistance to. Be an ideal situation to have would be a nice vaccine so that you can control the infection. No, so therefore, it is not that the people have not been trying this vaccine. They have been trying for many, many years, and various approaches have been used for leishmaniasis vaccine, starting from recombinant vaccines with and without adjuvants, like using proteins and peptides, DNA vaccine, using viral vectors to express those proteins, and also the whole parasite, inactivated whole parasite, with or without adjuvants. And, then live attenuated also has been tried, but those are basically chemically treated, radiation, or by passaging through the culture, and becomes the, so that the parasite gets less, virulent, and therefore used for this. But none of these approaches have produced a viable vaccine so far. So, but one has to remember, one of the strategies which was used in the past has been very successful, especially with the cutaneous leishmaniasis, where the term is called leishmanization, where you take a small little bit of, if a person gets a lesion, you take a little bit of that parasite from there, scratch the thing, put it, and then inoculate in another person, naive person, and that person then has a small lesion development, or maybe not a lesion development, but then they develop immune response, which is protecting for a long time, protecting for life in case. And this type of practice was done in former Soviet Union, Israel, Middle East, Iran, and other things. However, it has been discontinued because of the safety concerns. Because what happens is, you may take, it's not controlled process. You take a little bit, inoculating somebody. What if that person is immunocompromised and gets a full-blown disease? So obviously, under the current regulatory environment and under the current environment where we live in the world, this strategy would not be possible. So how can we mimic that strategy in a natural, in a way, so that we can use that similar kind of strategy? So the possible to achieve such a strategy is, us and others have thought about that if we can make a genetically modified parasite, which can infect the cells or infect the people, but will not cause a disease. So that's when we started this, getting into this genetically modified genetically modified live vaccines. First of all, it mimics the natural course of infection because it infects the people. It can, it's not dead parasite. It provides the complete array of antigens and because only thing what you have modified is taken out one or two genes, rest of all the proteins and everything is there. And then you have a control persistent. It can persist for a long time, because the reason leishmanization worked was that you have a low degree of infection continue, because you need that, in fact, a low degree of infection to create and have that protection, because that stimulates the immune system, which I will show you in a minute. And then when an infection occurs, it can prevent, it can quickly respond, and therefore the immune response can quickly kick in and therefore protect it. And also for the safety purpose, you know that these are, you know what has happened to the parasite. You have made certain mutations. You can nowadays sequence the whole genome and know from A to Z what is there and what is not there. So I think I will show you in a minute, define, and from the regulatory point, you need to have that well-defined parasite, unlike, passaging through the culture, irradiating, or chemically defined. You do not know where the changes have occurred. So this is just not to, this is a long, big, very dense slide here, but just to give you an idea that people have been trying. They have been doing in both in the visceral leishmaniasis, an agent like Danavani, then in L major, which causes cutaneous disease, Mexicana, and however, many there, and they have tested in animal models as preclinical studies, and mice, and it shows, and even in some primate cases, they show some efficacy, but haven't gone further much. So almost 10, 15 years ago, we started working on this, developing genetically modified live attenuated vaccine using a centrin gene deletion of the Aldanavani parasite. Now you ask, why the hell I picked up the centrin gene, and what is centrin gene? So the centrin gene, obviously, first of all, how did we pick up on that? We were at that time looking at the differential, because the idea was that what are the genes which are turned on during the infectious stage of this parasite? And can we look at those genes, and if we remove few of one or two or three, whatever we can do, can we make it a less virulent? And so in that process, we identified this centrin gene. But then it turned out to be the centrin is a very important gene, because it's a calcium binding cytoskeleton protein found in all eukaryotes. And however, and that it is localized mostly not mostly, it is localized in the centrosome in higher eukaryotes, whereas in the basal body in the lower eukaryotes, such as Leishmania. This centrin gene has been, people have worked out quite a bit and studies on that it is important for duplication, segregation of the centrosome and basal body, as well as associated organelles like kinetoplast in the case of Leishmania and the Golgi duplication. So we had found, we did a lot of studies, means Dr. Selva Pandyan in my laboratory did a lot of studies on this to define the function of the centrin. And there are 5 centrins in Leishmania, and each one has a little bit different function. However, one function does, if we disrupt one, it does not disrupt the other function. So when when Selva deleted the centrin gene from the Aldanavani and showed that what he found out, that we were thinking, when you delete it, the parasite will be, not divide or anything. Turned out to be, for us, serendipitously, lucky for us, that this centrin gene, which Selva deleted, does not affect the parasite division of the promastogote, which is the non-virulent form. Whereas when you take these parasites and transform into the amastogotes, these parasites do not divide. And they stay there for a long time and eventually die. So what happens? So when he looked at it carefully, he found out that these, amastogote, when they are transformed to amastogote, there is a nuclear division taking place, fine. However, the cell, there is cytokinesis is blocked. And once the cytokinesis is blocked, the lower part of the cell shows you that they become multinucleated, and eventually the cell, the cell death occurs. So that was good. So then we said, okay, let's try this vaccine, this parasite, L-Danavani centrin, in different models, just like anybody else did it. In the mice, we did and showed that it induces a multifunctional T cell response, which is Th1 protective response, which is good for leishmania, and also protects homologous and heterologous challenge against that is with this L-Danavani to L-Danavani or L major to L-Danavani. Then we tested it in hamster model, and because the hamster is as many of you know, hamster is the ideal model for visceral diseases. It mimics the disease just like the humans. It has splenomegaly, hepatomegaly, and weight loss, and eventual death. And so in the hamster model also, it protected. We also went in collaboration with Dr. Ricardo Fujiwara in Brazil to test in the dogs, and we showed when these dogs were immunized with this L-Danavani centrin, it developed a Th1 response, and when they were challenged with the virulent parasite, they were protected. And so basically, it showed that this vaccine candidate will work. However, when we were thinking about this thing, so the question, a lot of discussion took place internally, externally with our colleagues, with the regulatory bodies. Everybody suggested that they had a safety concern. Why? One, what if this parasite reverts back, and then also L-Danavani has a potential to visceralize. And therefore, and there is no treatment for that. Obviously you have to treat with the people with the drugs and all that. You don't want to do that. So that was a theoretical safety concern. Whether I will show you, and we showed that it doesn't visceralize or anything, but that was a theoretical concern. And then also the way these parasites were generated, they were generated by homologous recombination, where you put the antibiotic marker to select those type of parasites. So obviously these candidate had antibiotic markers, and neomycin, hygromycin, and at that time, there was no way to remove those genes, and therefore people felt that, because if you have a vaccine which has these antibiotic markers, if you immunize people, they will develop resistance to antibiotics. So at that time, with my esteemed colleagues, including Jesus and Shadin and Abhay Satoskar from Ohio State University, Greg Metaliskiski from McGill University, and also Shinjiro Hamano from Japan, Nagasaki University, we got together and thought, okay, if we can use a dermotropic, which is like Leishmania major, which does not visceralize, it will cause a lesion. Maybe if it will cause, but it doesn't go spread all over the different organs. And there are ways to control. You can put an ointment, as I told you, it is self-healing. You can heat treatment, and you can get rid of the lesion. So the advantage of this was that time we thought it does not visceralize, and the way we should develop as antibiotic marker free, and also should be cross protective. So therefore, we embarked on what we call it second generation leishmanization with a live attenuated markerless centrin deleted Leishmania major strain through CRISPR-Cas technology. So at that time, Greg in his lab had validated the CRISPR-Cas technology using other genes. So what he did at that time, he removed the centrin gene. So there are 5, as I told you, there are 5 centrin genes, and out of 5, the one which we had shown in L-Danavani, he used the specific primers for that, and by genome, he deleted that gene in L major centrin. We did the whole genome sequencing to show that there was no other gene it affected, including other centrin genes. Only the gene which we targeted, which is present on chromosome 22, has been deleted. And then, So by using CRISPR-Cas technology, and then he also, because initially you have to put the markers in, antibiotic marker. Then with the CRISPR, he got rid of those markers by serial dilutions, and so that they, this marker, this neomycin genes was out. And the reason we know it is out because we did the whole genome sequence and found there was no any antibiotic marker. The only thing was lost is this centrin gene, and which is shown on the Western blot here, that you can see that there was no gene, and when we add back, it is the centrin gene comes back there. On so when what we did was when Subir in my lab and Ranadir, they put these parasites into the mice and showed that the wild type shows an increase in as usual lesion formation. The knockout parasite had a little bit in the beginning, nothing swelling, and then eventually resolved after 6 weeks or so. Whereas if you add back to this thing comes back, Rorik. So which tells you that the centrin is an important gene for the virulence here, and which we had shown, nothing surprising here, we had shown already in the case of the Aldanavani. And when they looked at the parasite burden, both in the ear as well in the parasite burden in the ear where the parasite were injected, wild type you see quite a bit parasites over after 6 weeks or so, and then centrin hardly any parasites are there. When you add back, the parasite burden goes up. So showed us that the centrin, L major centrin knockout parasite behaves the way we do. So then we looked at whether this centrin gene is safe. Because it's important to know this parasite, L major centrin parasite deleted, is safe. So in Shinjiro Hamano tested, and Abai Satoska tested in various knockout mice, immune knockout mice, interferon gamma, STAT1, RAG2, and other parasites, and other mice, and showed that the L major centrin does not cause any lesions, and their parasite burden is very, very little. Then Ranadir and Subir, what they did was they artificially created immunosuppression in the normal mice. And by treating them, was immunizing them, then after 10 weeks, then they immunosuppressed them with the dexamethasone treatment, and after that, and then they looked whether the parasites grow back again or not. So what it shows you on the left side here, that wild type causes, you can see the huge lesion. L major centrin, there's no lesion. When you treat with dexamethasone, there is no lesion either. When they looked at the parasite burden, there were some mice who had some parasites in 1 or 2 mice which had some low level of parasite. So we wanted to know, oh God, have they reverted back? When Subir and Ranadir tested them by whole genome sequence as well as the Western blot, this shows you that the centrin gene was not there. This was basically some of them persist, and this is very important that these parasites persist, and therefore it's important that they will create an immune response for a long time. So now also in collaboration with Jesus and Chardin, what Ranadir did was that they wanted to know that whether these parasites are picked up by the sandfly, because if we immunize people, and if people are exposed to, when in the endemic area, they are exposed to infection. So what will happen is when sandfly may take this parasite up, then it has been shown previously that there can be a recombination between different parasites, and that's what I was telling you, that Aldanavani, the one which had a phenotype of cutaneous, was because of some recombination. And it is experimentally also shown by Dr. David Sachs' group and others that it can happen. But it's very low frequency. So therefore, the question was, can these parasites be picked up by the knockout parasites by the sandfly, and they will grow? So what happens is that when Ranadir and Subir did was that the hamsters were from which they had infection, which is the wild type infection and sentinel knockout parasite. When they fed them with zero diagnosis, that is, they fed the sandfly on these areas. What they found out that and followed them 2 weeks and 8 weeks, and found not followed them. They look at the parasites at 2 weeks of infection and eight weeks of infection, because by 8 weeks you can see these are huge lesions, and 2 weeks where bump a little bit, and there is nothing happening in the L major centrum. Immunized animals, and you can see that these sandflies pick up parasites in the wild type, but none picks up the knockout parasites, suggesting that they are safe. So then what they did was they immunized the animals. Because this was a one-step hurdle to pass before you immunize that. Are these parasites safe? Are they visceralizing? And they found out that only this parasite stays in this air, does not go into the liver, spleen, or other organs. So therefore, it was very clear that it is not visceralizing. Second, then they looked at the immune response, and in the mouse and the hamster, which is a standard model I told you earlier, and found out there was a strong Th1 response and multi-functional T cells. I don't want to go details in there because already this work has been published, and it just came last month in Nature Communication. So, and then they also looked at if we take these monocytes from the human, normal blood donors in the U.S., if we infect this parasite into these monocytes, would they induce an innate response, which is basically macrophage response? And they found IL-12 and a little bit of interferon gamma, TNF-alpha response. So suggesting that these knockout parasites do induce a good response in the normal human donors. So this is only an innate because these were macrophages. Then the question came up.
Professor Nakashi,
yes, please.
Can you please reshare your screen? Your presentation has actually gone small. We can't all see it properly.
Is that true? Because I think I have shared it.
So you will not see it, but someone's shared a screen in between. That's why this problem happened.
So what do I do now? You tell me what needs to be done.
So you need to present now, I think, again. Maybe.
Okay, present again. How do I do that?
So, you would have it, that present now option, right hand side corner.
Do I have what option?
Present now option. I mean, is it visible to you?
I don't think I can see anywhere.
Can you go to the bottom right of your screen?
Yes.
I am at the bottom right of the screen.
Maybe sir, you can skip your presentation and redo the present now. Maybe that should help.
Oh God. All right. So stop sharing then.
Yes, and then maybe you can again share back with the same slide.
Okay.
Or if we tell someone called Anindita Bhaduri has presented it. It was shown in
yes, sir, I did. But I think her screen is gone. But
okay. I was getting the screen perfectly. Anyway, some may not get. Let him be.
I cannot. I don't know where to. Oh, okay. Now, okay. So.
Sir, you need to pin it, sir.
Resume your present. Should I do resume your presentation?
Yes, sir. You can and pin it to screen.
Your screen is still visible to others. It says, click to cancel your presentation.
Maybe that should help, sir. You can go out and then come back. Also, you can pin the screen. You see there's a bell kind of an icon.
Okay, stop sharing.
Yes, sir. You can start sharing.
Okay, now present now.
Yes, sir.
Okay, your entire screen. Okay. Share. Can you see now? Okay. Okay,
yes,
yeah.
No, it's back, sir. It's fine.
Okay, all right. Okay, great.
Okay, thank you very much.
Thank you, thank you. God, I am not good at this technology, as I told you. So forgive me for that. So, all right. So, what I was trying to tell you that so everybody can see it now fully.
Yes, sir.
Yes, sir.
Okay. All right. So once we developed this candidate vaccine, we showed that it is safe and immunogenic. The next question comes: is it protective? And it is efficacious in a regulatory jargon. But at that time, Nathan Peters and others had shown that many of these vaccine candidates, which were, as I showed you the list of those vaccine candidates, when they usually challenged the animals with this needle challenge. They were protective in the needle challenge. However, when he and others tried against the sandfly-mediated infection, many of them did not show a good protection. So the question was, what is there, which you will hear quite a bit from both Jesus and Sharon, what those factors are. I'm not going into detail here, but what there are many other factors which are important for this sandfly-mediated infection, such as the microbiota, the PSG gel, the other exosomes, salivary gland proteins, all that. Then when the sandfly takes a blood meal and injects the parasite, it injects a lot of other components. So therefore, that modulates the immune response quite a bit, and you will hear in detail that from Jesus and Sharon. Okay, now, what do I do? How do I move to this thing? Oh God, this thing is... Am I stuck?
Hira, you're making me nervous.
I know, because, this thing is, I haven't very little bit done with this Zoom business. Not Zoom. The Zoom is a little bit better than these. I don't know where to find this thing now. I cannot. So how do I change the next slide here?
Yeah, sir, guys? You have to go to the PowerPoint presentation.
Yeah. Okay. All right. I'm okay. This. Okay. All right. Sorry.
That's fine.
All right. Okay. Sorry. Learning. Jesus and Sharon, now you know what to do. I'm paving the way for you guys, all the mistakes which I'm making here. All right, so then what Ranadir and Subir did was that they challenged these mice, which are immunized for 8 weeks and immunized for 8 weeks, and then challenged them with the sandfly infection and followed up to 10 weeks. As you can see that, naive, which did not get anything, you have no lesions. Non-immunized, you can see almost the air is falling apart. And then immunized challenge, it is protective. And as you can look at the parasite, air thickness, there is not anything, and the parasite burden was also very little. Then, so this was against the Leishmania major through sandfly infection. Then they tested Leishmania donovani through sandfly infection, which is a gold standard in the hamster model. So what they found out that you can see those which were non-immunized, they lost the weight, whereas those which were immunized challenge, and they did not lose any weight. This is a normal weight. If you take a normal hamster at that particular age group, it is 160 grams, it was 100. You can see there's a weight loss. In addition, you see splenomegaly, non-immunized challenge, you can see huge spleen. Immunized, it is like a normal, non-infected, not immunized animal. And then you look at the mortality over the 14 months, you can see these immunized ones survive, all of them, whereas non-immunized ones start dying one after the other. And then lastly, there's a control of parastemia. The wild type infection keeps on going, whereas in the case of the immunized challenge, the parasite goes down both in the spleen and the liver. This is the L. donovani parasite. infection. Now, so having said that, in next 15 minutes or so, I will talk to you. Now, since we showed this L major centrin can both protect its homologous challenge and heterologous challenge, the question is, as I said, is this similar to leishmanization? Only and the difference is that in leishmanization you may have is a safety concern, whereas here we showed you that there is no safety concern. So we compare the immune response against the live attenuated centrin against the leishmanization. So how did we create leishmanization? Leishmanization, basically you give a low dose of infection, and then over a period of time they develop lesions, such as here a little bit, and then they, as the parasite, the mouse recovers, and then you don't see the clearing of the lesion. So that means at this stage, now these animals are leishmanized in the sense they have recovered from the infection, and they hopefully are protected. Whereas in the case of our centrin, you can see over these 12 weeks, they don't develop any lesion. So at that time, and we will measure the immune response, and you can see the immune response and the T cell immune response basically is similar in either if you do the wild type primary infection, which is the healed one, or with the immunized challenge, leishmania. So then at the next step, we then, can they recall the response quickly as leishmanization has been shown to have? So that is our, the L major centrin. So you can, after 20 hours, they infected the animals with the wild type infection, and you can see there is the mediate, T effector cell response. These are the effector cells and producing interferon gamma, both leishmanization as well as in the case of the immunization. And the total, increase is similar between the wild type with the leishmanization as well as the immunization. Then they ask the question, if you infect at this point after immunization with sandfly, after 5 weeks, would they control the similar kind of a parasite burden? And what they found out that if you look at the air parasite burden in the air and draining lymph node, they are similarly controlled, whereas in uninfected they are not. So what it told us that this leishmanization, L major centrin parasite immunization is equivalent to leishmanization. And also, not only because after 5 weeks they can protect, in this case, or 8 weeks immunization, this is a long-term protection. What we showed that if you immunize these animals, and after 36 weeks of post-immunization, which is almost 9 months, then you challenge them with the parasite, and you can see that they are protected, both immunized and leishmanized animals. Whereas in the case of naive, you can see starting the lesion development. So then, having established that this parasite is, good vaccine candidate, safe and efficacious in the preclinical model, and then to also it is equivalent to leishmanization, but it is a safe alternative. The next question comes: how do we take this parasite to the bedside, to the people, and in that, what is involved in that process? So in that process, it is obviously you do basic research, which I have been talking for the last 38 minutes, that you do all the antigen vaccine development, validation, do a little bit of human immunology to show whether they are, these human PBMCs or monocytes are responding to it. And then once you have done several reiterations of that, you then go to the GMP vaccine development to develop a vaccine under GMP condition, which is safe, stable, and then you can go to the clinical trials. So in that process, we were lucky to have the collaboration with Dr. Sanjay Singh in Genova Pharmaceuticals and Genova Biopharmaceuticals in Pune, and who is himself a very good, renowned immunologist as well as vaccinologist. And he was kind enough to collaborate with us to develop these parasites first we developed under GLP conditions, which are, those GLP conditions are CGMP compliant approach. basically taking from a frozen stock to a bioreactor and looking at the parasite growth and the density. So basically, he was able in their bioreactor, a small scale, 1 liter scale, to grow these parasites very nicely. And these parasites, as you can see, he shows you that it is in the wild, ready to be vaccinated. Obviously, there's much more to be done yet. And then when he provided us that GLP grade vaccine parasites, and in our lab, what we did was, again in collaboration in the hamster model, and see GMP compliant shows a similar kind of immune response as a lab grown, because lab grown conditions are a little bit different than the GLP, and you can see they in the spleen, and you can see similar kind of immune response, the interferon gamma, TL alpha. And then when they were challenged, after 6 months of challenge, you can see this GLP grade material protects, controls the parasite burden, as I had shown from the lab grown parasites previously. So suggesting that this GLP material is comparable to lab grown and can be ready for further characterization. And what we did in collaboration with Dr. Pradeep Das and Sushmita Das in the RMRI Patna, and they took these, and they infected this GLP grade material in the healthy in the population PBMCs from a healthy population of an endemic area, because they had shown that these people are RK13 negative, which is indication of the previous infection, and showed that you can, in these PBMCs, you can induce a very good interferon gamma response and a little bit of IL-10. But when you look at interferon gamma IL-10 ratio, you can see it is much higher. So suggesting that these major parasite GLP grade material are capable of inducing the immune response in the healthy population in the endemic area. Studies are going on to see whether heel visceral leishmaniasis and other population, how that immune response is changing, and also so in the PBMC. So with that, and I want to summarize the thing that what I told you all in the last 42 minutes, developed with a marker-free Leishmania centrin, and through the CRISPR-Cas, it is safe, it is immunogenic, it is efficacious, and it is comparable to leishmanization. It can induce both immune response and pro-inflammatory response in the healthy people, both in the non-endemic and endemic situation. And and we are now ready to get into the GMP making GMP material, as well as the toxicology studies. So basically, the next step is to make a GMP grade. We are lucky to have funding from this Japanese group, and Genova is poised to develop this pattern in GMP grade. Then we are going to do the toxicity studies as well, and then obviously we will also have, we are planning to have phase 1, phase 2 clinical studies in US and endemic areas, especially in Ethiopia and India, and also in the US. So that's the plan. So with that plan, I want to thank you. But before I thank you for listening, I want to acknowledge the people who have helped us through this whole journey. As I mentioned, while I was talking, people in my lab, Ohio State University, McGill University, Nagasaki University, RMRI Patna, in Brazil, and of course, my colleagues in NIAID, Shadin, Jesus, and David Sack, Genova, and then obviously these are the planned studies, proposed PS1 phase 2 in both in US, Ethiopia, and India, and also the earlier studies, which I forgot to mention. It was the Al-Danawani studies were initiated by first time with Dr. Poonam Salotra's group in Institute of Pathology in India. So with that, I think I want to thank also the funding sources, the FDA intramural funding, NIH funding, the Global Health Initiative Technology Fund, which is the JET fund from Japan, as well as the Wellcome Trust. We are also testing this vaccine candidate in the dogs in Tunisia, and we're hoping that when exposing to the natural infection, and those studies are underway. With that, I will stop and take any questions. Thank you.
Thank you. Thank you very much, sir, for your illuminating lecture. Now, I would like to hand over the session to Dr. Aritri Dotto. She basically did her PhD in our institute, in our laboratory, and now working as a postdoctoral fellow for in CCMB. Please, Aritri, please take over the question and answer session.
A very good evening to everyone. As Professor Pal mentioned, looking forward to the questions for Professor Nakashi. So, Professor Nakashi, we have the first question. So, the question is, what are the role of co-inhibitory molecules in Leishmania donovani, in Leishmania vaccine-induced immunity?
Co-inhibitory molecules. Yes, we published a paper several years ago, CD200, and they, as well as the what we have seen is that also the other co-inhibitory molecules, they have these vaccine candidates suppress those co-inhibitory expression of co-inhibitory molecules and induce the co-stimulatory molecules, and therefore that we have studied that, and I think the answer to the question is they inhibit the expression of co-inhibitory molecules.
Okay, so the next question is, what is your opinion on whole-killed Leishmania vaccines as potential vaccine candidates?
Well, people have tried that whole kill parasite with and without adjuvants, as I mentioned earlier. However, when they went into people, and it was not successful. It was tested in Iran and other places. However, there was the problem could be that you need, as I mentioned to you earlier, it is not the antigen alone, and obviously, if that was the case, so we would have a vaccine by now, where you have done all sorts of antigen. Heat inactivated or kill parasite is basically you provide antigens, and then you also add, in some cases, the adjuvants to boost the immune response. However, those things, if the adjuvants and the antigens itself were important, were necessary, we would have gotten a vaccine by now. But what I said was the biology of the parasite tells you that from our experience with Leishmanization, you need to have some kind of an infection, which the parasite will not have. And in my opinion, people tried and didn't find it, but I think that's why we approach this other approach of live attenuated parasite.
Okay. So the next question is, what are the implications of T cytotoxic memory cells in vaccinated men?
Can you repeat the question, please? Sorry.
What are the implications of T cytotoxic memory cells in vaccinated mice?
Well, we have not looked at it, and I cannot answer that question at this time, but I think we need to look at it. And what we looked at it, the effector cells, the memory cell, we have looked at the memory response, and they generate a good memory response. And but we have not specifically looked at the inhibitory cells.
So the next question is, why it is named as second generation Leishmaniazation vaccine? Is it effective for visual Leishmaniasis also?
Well, of course, the reason we call it second generation Leishmanization is because Leishmanization was always done with the live parasite. Okay, and so with this with this live attenuated parasite, which does not cause any lesions or anything, we call it second generation. And what was the second part of it?
The second part was that is it effective for visceral leishmaniasis also?
Well, I showed you the data in the hamster model that it is, because if you immunize the animals with the hamsters, and in fact, maybe I was not clear, sorry about that, and then you infect them the sandfly mediated L. Danavani infection, you get protection. So yes, the answer is yes.
Okay. Sir, I have a question. I was wondering, why did you choose the 37-week immunization? What was the standard of choosing that?
Well, nothing specific, on an auspicious day or anything else. But it was that we wanted to look at as much long we can go, because after 12 months or 14 months of animals, they don't, especially the mice, there's a lifespan, and so the 9 months translates into many years in human. So I think we wanted to. It was more of convenience, but just to show how long you can go there, and therefore you protect. And these studies were done by our colleague, Dr. Shinjiro Hamano in Japan, and so he's selected, and that was the extreme end at that point, and he wanted to get rid of the animals after that, so he just looked at it. So nothing specific.
Okay, sir. So there's another question. Why does the patient of visceral leishmaniasis suffer anemia, although this parasite does not infect RBC?
That's a good question. I'm not a clinician. I cannot answer that. So I'm a basic biology person, but I think maybe somebody else may know about. But I'm sorry, I don't know why it causes anemia.
I do not see any more questions. Thank you, Professor Nakashi. I would hand over back the mic to Professor Pal, or if there is any other questions. I don't think so, sir. Professor Pal.
Thank you, sir. Thank you, sir, Dr. Nakashi, for your kind consideration and approval to talk as an expert in the platform of our West Bengal State University. On behalf of West Bengal State University, I would like to convey our gratitude and thankfulness to you, sir. Thank you very much.
Thank you. Thank you very much. Okay, now how do I stop sharing? Okay, so I should, okay.
So, may we please go to our next session? Before that, I would like to request all the audience, please mute your microphone and also your camera, please. So, now I am presenting Dr. Shaden Kamhai. Dr. Shaden Kamhai received her PhD in medical entomology from Salford University, England in 1990. Dr. Kamhai was an associate professor at Yarmouk University in Jordan before coming to NIH as a visiting scientist. She is currently a core associate scientist at the Laboratory of Malaria and Vector Research at National Institute of Allergy and Infectious Diseases, NIH. Since coming to NIH, Dr. Kamhai worked on vector competence of Leishmania in vector sandflies and established the first models of pathogenesis following transmission of Leishmania by vector mice. This ignited her interest in understanding the virulence and vector-transmitted pathogens through investigating vector and host-derived factors that influence the parasite establishment at the site of infected vector mice. Dr. Kamhi continues to collaborate with several international partners, including colleagues from the Indian subcontinent, to validate the relevance of her experimental findings under natural conditions. She has published more than 100 articles in her credit. So, please, Dr. Kamhi, I welcome you in this webinar series.
Thank you, Chiranjib, for your invitation. It's a great pleasure to be with you all. And good morning to whoever is in the US, and good afternoon to people in India. So let me tackle the technical parts first. Okay, let me see. Can I put the presentation in presentation mode now?
Yeah,
right?
yeah. You just open the PowerPoint slide and then go back to the Meet online platform. Extreme right corner, you have a present now.
Yes, I'm doing that. My question is, can I put it in presentation mode?
Yeah, sure.
Okay. Yeah, it is coming up, Ashadhan. Yeah. So just you go to this present now. Yeah, you can.
Can you see it?
Yeah.
Yeah, I can see it, but it's miniature form. So can you?
Yeah, that's
okay.
Now.
Perfect.
All right. So this talk I know you're all joined to hear about vaccines for leishmaniasis, and Dr. Nakazi has given a great lecture about one of our most promising vaccines. But if you listened carefully to what he was saying, in fact, he said several times how important it is to challenge these animals with sandflies that are the natural vector of the leishmania, because it's the gold standard. And so my lecture really here is to show you why that is, why we have a virulence of sandfly transmitted leishmaniasis, and that it is the outcome of a complex interaction between many vector-derived factors and our own host immune system. So this is just to remind everyone that for the most part, apart from some incidences of vertical transmission that are mainly in dogs, stable leishmaniasis is only transmitted by the bite of a phlebotomous sandfly or a Lutzomyia sandfly. And these sandflies can transmit multiple forms, from cutaneous to visceral. And what I want to stress here is, even for vaccines, why we need to understand vector transmission. Why does it matter? And Hira has already alluded to the fact that there is the famous paper from PLOS Pathogens that vector transmission of leishmania abrogates vaccine-induced protection against needle challenge. That means if you are satisfied with just a needle challenge, you come and you challenge your animals with a sandfly bite, you find that in fact it doesn't work. But less known are other publications where the reverse is true. For example, Matt Rogers published a paper showing that synthetic glycovaccine did protect against bites of infected sandfly, but failed against the needle. And Jesus is going to elaborate a lot more about some work our group has done, where we have shown that salivary molecules can protect and act as vaccines. And we only have gotten to that by knowing that saliva is important in modulating the response of the host. But if you take all of it together, these kind of suggest or indicate really that the environment, when you challenge the host with an infected bite, is very different. It's just qualitatively different from just a sterile injection of the parasite, despite the fact that it's the parasite that goes on to develop the disease. So why is that? So here has already showed this image from a chapter we wrote, and this is just to show you that the sandfly is a biological vector of leishmania. The leishmania parasite goes through several complex developmental stages as it goes from the posterior midgut all the way to the anterior. And that can take between 7 to 15 days. So it's quite a long life cycle, and the parasite interacts closely with multiple components of the midgut. And then it itself starts to secrete molecules that in this small confined place will start to accumulate. And as Hira mentioned briefly, some of the most known are the promasticode secretory gel that is shown here in yellow. And this is a very soluble gel-like and sticky substance made of filamentous phosphoglycans. And that captures everything in this anterior midgut, that when the parasites comes to transmit, the infectious inoculum, what I call now infectious inoculum, is not just the parasite, but several components that are crucial to allow the parasite to establish the infection in the host. So what are these? So far, and I'm sure that in the future, our research will lead us to more, what we of course know that we need the Leishmania metacyclics, the infectious stage. We have the promasticode secretory gel, that sticky substance at the anterior that is also transmitted. We have Leishmania themselves that secrete exosomes, and there's been many studies of the secretions in culture, but we've shown that it secretes it in the sandfly. And of course, there is saliva that the fly, infected or not, needs to put into the host in order to allow for feeding. And of course, these include very potent molecules like vasodilators that permit the blood to flow faster, anticoagulants to stop the blood from clotting, etc. And of course, we have a healthy community of gut microbiota, and we've done some work that I won't present here to show that it's very important for the parasite to develop in the fly. So we have all these components, and actually each of them have been shown to enhance leishmaniasis in various ways. And in this talk, I'm going to focus on the more recent studies that we've done in gut microbiota, then lead you to further unpublished data that came out of this study as well. But here, just to show you, this is like images by electron microscopy from the section of the gut. And this shows you the cell body of a parasite with an exosome where the red arrow is that's budding off it. And they form multivesicular bodies that we can also see being secreted. And if we look at anterior guts of mature infection where the parasites has really accumulated, you can see from the arrows how many more exosomes end up being in that niche. And we did a study that's published in 2015 with Miguel University and Martin Olivier, where we show that actually these exosomes are immunomodulators, and if we inject them, the classical experiment with parasites, in this case, purified metacyclics, you can see in black that alone the metacyclics do develop a lesion, but it is actually significantly increased when we add the exosomes. And these were actually gut-derived exosomes from the sandflies. That was quite laborious. And also, if we look at the pathology, we can see that the lesions are much more virulent, or the pathology is much worse. And of course, this is reflected by an increase in parasite load. So then these elements are foreign bodies that we inject into our skin. So the immune response of the host will respond to anything foreign. So it's not a surprise. And in fact, if we replace exosomes with saliva or with the promasticode secretory gel, we can more or less get the same profile, an increase in lesion size, increase in pathology, and increase in parasite load. But in fact, these are artificial because we are injecting them alone. We're injecting them in volumes we choose to be representative. We inject them in volumes that are huge for compared to a sandfly bite. I mean, the usual minimal volume you inject these things in is about 10 microliters, which is huge compared to the probing of a tiny proboscis whose diameter is only 0.1 micron or less. So we wanted to explore what is really happening just at the site of an infected sandfly bite. And before we started our study, there was one also very famous paper from David Sachs's group that showed, in fact, that neutrophils that are very important for parasites establishment, because they hide, the parasites hide in neutrophils as Trojan horses till the macrophages arrive. And what they showed, and did not explain why, is that the neutrophils at the site of the bite persist for a long time, and they did it in a very elaborate live imaging. And what you see here is the persistence of these neutrophils in the skin for up to 60 minutes. So when we started to look at the bite site, and I did this project mainly with Ranadir Day, who's actually part of Hira's group, and it was a great collaboration, we decided to look at a model of visceral leishmaniasis that has previously was not actually done before, and we used Lutzomyia longipalpis that can grow and transmit Leishmania donovani very well. And then we looked at what happens in the simplest way possible with the least manipulation at the site of the bite. And what you can see here is, again, as David Sachs and Nathan Peters found, we do see a far more intense accumulation of neutrophils at the bite site compared to injections of parasites alone. So the first panel shows you 3, 6, and 18 hours after an infected bite versus injection of Leishmania by itself. This is just to simplify. We did many other controls that you can see. This paper was also published in 2018. And then doing a very targeted inflammatory panel at what's going on at the site of the bite, just to give us some clues. You can immediately see that these are all our control groups, actually. The first two are infected and uninfected bites, and the other two are when we inject Leishmania with saliva or salivary glands, or Leishmania alone. And it's striking how different the early inflammatory response to the bite that are similar, much more similar compared to the injection of the parasites in whatever form. And we knew, for example, that CXCL1 is the canonical chemokine to attract neutrophil, is upregulated, CCL2 for attracting monocytes, YM1 to turn them into NM2. So a lot had been known in the background. But what struck us from this study is that IL-1 beta, which was never really explored in the context of a sandfly bite, was consistently and highly upregulated at the bite site, more than 1,000-fold higher than in naive skin, and more than 100-fold higher than if we injected Leishmania alone. And this panel just shows you the timeline from 3 to 18 hours. So we asked the question, why is that? And what is actually producing the IL-1 beta? So we simply took the ears 6 hours after an infected bite, recovered the cells to do flow. And what we can see is the majority of these cells that are producing IL-1 beta. So here you can see on the left, just simply the IL-1 beta producing cell against forward scatter. And once we identify the IL-1 beta population, we backgate it to see what kind of cells they are. And they are LY6G high, LY6C intermediate, and CD11B positive. They're definitely neutrophil, and 86% of the IL-1 beta comes from these neutrophils. So basically, they're the main source of the IL-1 beta. And IL-1 beta is a very powerful inflammatory cytokine and is regulated at many multiple steps. And so the canonical pathway to the production of active IL-1 beta is through inflammasome activation. And what you see here is a Western blot that Ranadir optimized from just simply the lysate of the ear. And what we can see here is, if we look at just naive skin compared to infected bites, you can see, and here we chose to look at the NLRP3 inflammasome because it's the most promiscuous, responding to the widest signals. And you can see it's upregulated after infected bites. We can see pro-IL-1 beta. And also, most importantly, we can see a lot more expression of procaspase and cleavage of the procaspase to caspase, which is really the penultimate step before the pro-IL-1 beta is cleaved to be released from the cell and to be active. So this suggested that indeed the IL-1 beta is coming from activation of the NLRP3 inflammasome. But since we struggled to show the actual active IL-1 beta, which is about 17 kilodaltons from the Western, we wanted to show that at least the inflammasome complex has been formed. And what we simply did is we immunoprecipitate with the antibody against ASC, which is the scaffold adapter protein where all the elements of the inflammasome dock in order to form a complex that can cleave caspase. And then we probed it with NLRP3, and you can see that the complex is formed after the infected bites. So where we are now is, in fact, we know why these neutrophils are coming in and persisting now, because they secrete IL-1 beta, because they have an activated inflammasome, and that IL-1 beta actually acts as a powerful chemoattractant for more neutrophils that then causes this active and potent autocrine loop. So where is it coming from? Obviously, the canonical trigger for inflammasome is through toll-like receptors and bacteria. And so we know that the microbiota is very rich in the gut, and we had just finished a project characterizing the microbiota. And so what we did is a simple experiment. We took the infected flies in a container with a meshed net, and we overturned LBA agar that's been warmed on them and allowed the flies to probe. And there are many other details in there as controls. You just have to read the paper to go through them. And what you can see here is indeed we see some bacterial colonies growing from within the feeder, but really not outside. But as a second confirmation, we actually could amplify the Leishmania from these growing bacterial colonies. So knowing that these bacteria were Leishmania positive and came from the fly, we wanted finally to identify them and match them to the microbiota in the gut. And then with 16S sequencing, we could really find a beautiful match between the Leishmania positive probe site colonies and from these, the guts of the flies that did the probing. So that kindly clinched or closed the speculation that the microbiota is indeed egested, and this is the cause of this huge pro-inflammatory surge that occurs and the really persistent and intense infiltration of neutrophils to the bite site. So why is that important? So we did two experiments from two different angles. We cleared the gut microbiota by antibiotics. So we gave them antibiotics that do not affect the parasites. So all these controls have actually been looked at. And then we allowed these flies that were infected, half not given antibiotics, half given antibiotics to transmit. And what we can see strikingly is that the intense infiltration we expect to see, that's like the hallmark of the bite 6 hours after Leishmania transmission, have been really abrogated by the antibiotic treatment of the flies. And what was also striking is that when we looked 3 weeks, as early as we could think we could detect parasites in the spleen, we could see a significant reduction in the number of mice where we could detect parasites that actually made it from the ear into the spleen after transmission. And just in case still something is happening to these antibiotic-treated flies, we did another approach. We actually used anakinra, which is an IL-beta blocker, So we gave that anakinra to the mice, so the sandflies are untouched, and so the same sandflies transmitted to the mice. And what you can see here is, again, at 6 hours, we see the same thing, a complete abrogation of the neutrophilic infiltration that we see at 6 hours. And since this was really cleaner, because anakinra is a potent blocker of IL-1 beta, we really blocked the ability of the parasite to go from the skin to the spleen. And in fact, at least to my knowledge, this was the first single mediator that we could point at that says it could really block parasite visceralization. And we're following up on this data to understand more the mediators of L. donovani and other visceralizing parasites pathways from the skin to the organs. So this is like in summary of that first that has been published work, which is that we showed that ingested mid-gut microbiota adds to the infectious inoculum. It primes the NLRP3 inflammasome and produces high levels of IL-1 beta, and that these act in an autocrine manner that is responsible for the persistent recruitment of neutrophils to the bite site. And this overall outcome, in fact, leads to more neutrophils, and the neutrophils attract more monocytes. I haven't shown it, but we looked at that too. So you can imagine that just from the bite and from having the microbiota, in addition to all the other things, you do get a very hyper-inflammatory milieu at the bite site. That would not be there if you injected the parasite without anything else. So that led us to ask the question, which is quite a simple question to ask, but you need to think of it, is how do these parasites survive this inflammatory assault? Because if this continues and this loop is like autocrine loop, what's stopping it? Because ultimately it's going to hurt the parasite. So we have two very talented postdocs that really worked on the second project that's still unpublished. And we focused on what's going on, what other things could possibly be going on. And this really showed us that there is a contribution of host factors that is equally important to pathogen establishment at the site. And kind of points to the fine-tuning of the parasite using both vector-derived factors and also host responses to make sure that it establishes well. So Tiago and Eva did a lot of work that I'm going to show you here. And when we think of vector bites, many, including ourselves, really focused on the typical leukocyte infiltration, the innate cells that are involved in establishing an immune response. And of course, this begins with neutrophils because in tissue injury, it's an innate, conserved, very old pathway that is the wound healing response with very defined phases, and its point is to restore homeostasis, resulting in all these leukocyte infiltrations that has been studied in the context of the pathogen. But another not so obvious but very prominent outcome to a vector bite is due to tissue injury, we will have breakage of the capillaries because that's what the sandfly and even other vectors want to do. They want to destroy the capillaries, get the bloods out of the capillaries so that they can ingest it. And if we have this red blood cell leakage, then the host must clear it to restore homeostasis. So I'll tell you a little bit about that part. First of all, just let me show you that we simply did H&E sections of the site of the bite to show that indeed, compared to a naive skin where you can see the black arrow, this is simply two magnifications, where the blood cells are really contained in the vessels. 6 hours after the bite, and still at 18 hours, we get a lot of leakage from these hemorrhaging capillaries into the tissue. And this is really not good for the host. So what does the host do? So when everything is fine, heme is usually bound, and it's the protostatic group that is bound very tightly to hemoproteins. Of course, the most famous being hemoglobin, that is a major component of the red blood cells, as well as other proteins. So what happens if heme comes out? So for many reasons, actually including harsh inflammation can induce this, but you can get the RBC, if it's damaged somehow and releases this hemoproteins and its heme is highly toxic to the tissue and results in a well-known Fenton reaction that produces radical oxygen species that then promote more inflammation and resulting in really tissue damage and cell death eventually. So it's really something that the host would like to control, and therefore these cells have produced a cell stress response that actually will block the Fenton reaction. And it does it by catabolizing heme, removing it from the environment. And it does this by this enzyme called heme oxygenase 1. And heme oxygenase 1 is an inducible cytoproductive enzyme that catabolizes or catalyzes the oxidative degradation of heme, as you can see here, whether from heme proteins, releasing heme, you get heme oxygenase actually catabolizing it to produce iron. That is important for the cells. It reduces as a byproduct carbon monoxide, and I'll talk more about these later, as well as biliverdin, that's automatically reduced to bilirubin. So this heme oxygenase 1 is something I've not heard before I started thinking of blood and RBC leakage. And in fact, it was a colleague, Valeria Borges from Brazil, who works on it as a risk factor in visceral leishmaniasis, since it's increased in the serum of patients and since it has anti-inflammatory roles. But just the minute she was talking to me about heme oxygenase 1 and that its substrate, its more potent substrate is heme, I was thinking, well, what's happening to those RBCs that are being leaked to the surrounding tissue? So with Valeria, we looked at the bite of Lutzumia longipalpis, and this is also published in 2018. And we use the spleen because that's a site where a lot of old and senescent damaged RBCs are recycled. So it has a lot of heme oxygenase as our positive control. And if you look at the timeline after the bite of Lutzumia longipalpis, you can see a very strong induction of heme oxygenase 1 at 24 hours and 48 hours. So then that is how the story begins. We do see heme oxygenase induction after the bite. And heme oxygenase can be induced by many things, including IL-10, but its most classical pathway is through the transcription factor NERV2. So we used knockout animals. This is just technical to show the pathway, and it's actually NERV2 dependent expression of heme oxygenase. And then we used flow to sort the cells from the bite site in the ear to see what is producing this heme oxygenase out of the cell population. And in this early study, we actually showed that classical macrophages and to a lesser extent in this study, inflammatory monocytes are the main producers of heme oxygenase 1, not non-myeloid cells, not neutrophils, but the macrophages actually. And we measured it by ELISA as well, and you can see that after the bite, Lutzumia longipalpis really produces a lot of heme oxygenase that's highly significant against naive skin and against needle pokes that we used a very fine needle to just poke the ears about 10 times. So this is not just Lutzumia longipalpis. We have another sandfly called Phlebotomus dubosci, and this transmits Leishmania major, while Lutzumia longipalpis transmits Leishmania infantum. Unfortunately, we do not have a colony of P. argentipes, although we'd love to get one. To do similar experiments. And here again, you can see compared to needle, we do see induction of heme oxygenase 1 by the two vectors. So going back to the spleen, since now we know that we have macrophages that are producing hemoxygenase, there are in fact professional erythrophagocytes in the spleen, and they express certain markers. The two most known ones are CD163 and CD91, and these are receptors that actually capture the complexes between hemopexin and haptoglobin and heme or hemoglobin, so that they can internalize them into the cell. Of course, they can also gobble up the whole RBC and then break it down. Basically, inside the cell, then the heme is, the presence of the heme is actually, after proteome degradation, activates hemoxygenase induction. It removes repressors, adds activators to allow a truncated hemoxygenase to move from the cytoplasm to the nucleus and become active. And there it does its reaction to neutralize heme, producing carbon monoxide and bilirubin, and the byproducts are gaseous, meaning they can infiltrate, go out of the cells. And in fact, many studies have shown that they have potent antioxidant and anti-inflammatory properties. So that is well known in the realm of spleen research and red blood cell senescence. But what about at the site of the bite in the skin? There has been, to our knowledge at least, nothing known about that occurring in the skin. So the first thing we did is again just focus on the population of myeloid cells after bites from the skin. And the first thing we looked at is, could we see any cells actually taking up RBCs? And you can, we use this antibody called anti-TER119, because this is a marker that is present exclusively on the surface of red blood cells. And so when we take the myeloid population and look at the production of TER119, you can see that at 18 hours, which is around the time where we begin to see, remember, hemoxygenase in our timeline peaks at 24 to 48 hours, you can see that there is a clear population, about 9% of these myeloid cells that are internalizing TER119 transiently. And then when we actually quantified these from many experiments for both myeloma and phlebotomist Bosky, we found that indeed there is an increase in the number of cells that are positive for TER119. So this is just simply showing you the number of CD11b positive, TER119 positive cells. And then when we also backgate to see what are the TER119 positive cells, we can clearly see that they are mostly macrophages that do express the marker F4/80, and that some seem to be resident macrophages that are LYC negative, while others may have a monocyte origin, since they are intermediate for LY6C. And that's in both of the vectors. So what this tells us is, in the skin, we do find internalization of RBCs by macrophages, and the timeline fits when they actually induce hemoxygenase 1. So we said, well, they're in the spleen. Let's see, do we see this population also in the skin? And what was remarkable is, even in naive skin, we see a very strong subset of cells that are expressing these two markers, the CD91 and CD163, indicating that the skin compartment also has standby resident macrophages that are professional erythrophagocytes. And these are increased, but not significantly after the bite. And then when we actually gate on these macrophages, you can see that at naive, there's no TER1 positive cells. Most of the RBCs are within their vessels. And here, when we look at 18 hours after the bite, we see our population of macrophages that are gobbling up the RBCs, but we also equally see this population without RBCs. And this indicated that we do have a resident population of these professional erythrophagocytes. And to further prove it, we actually did some confocal microscopy that really took a lot to optimize, because it's really looking at the ear tissue and skin, which is tough after the bite, after a sandfly bite. And we just looked at several panels. And this is an orthogonal slice, so you're going through by depth. And what you can see in yellow are macrophage marker, IBA1. And you can see that the RBCs, the TER119 is in white, is internalized inside the cell, and that this cell, through DAPI and hemoxygenase 1 in red, is strongly positive for hemoxygenase 1. And this is the same for all the other three panels. And what was also obvious, if you look at the green arrows, is that the neutrophils that we also stain for with myeloperoxidase do not express hemoxygenase 1, as we found in our previous study and with the flow. So this tells you that at the skin, we do have these erythrophagocytes that are internalizing RBCs and are breaking it down so that it becomes and produces hemoxygenase. So what is the actual outcome of this hemoxygenase? We collaborated with Miguel Suarez in Portugal, and he actually sent us hemoxygenase knockout animals. And we simply infected sandflies and then transmitted to the knockouts or wild type, and we measured the lesion and looked at the parasite classical. So the first thing that we could see is, in fact, in the absence of hemoxygenase, and that's in red, that's the hemoxygenase knockout, we see a significant increase in the pathology of the lesions. But what was surprising to us was that, in fact, there was not a major effect on the parasite load. And that is really the definition of disease tolerance, induction of tolerance, because tolerance is a way by which the host actually protects itself from pathology, but in ways that do not affect the pathogen necessarily. But in this panel, we used the hemoxygenase knockout. And to me, that was highly problematic, because these animals do not produce hemoxygenase at all. And I really wanted to mimic a transient suppression of the hemoxygenase around the same time as we see its induction in the fly, and see if it has any effect. So we used a specific inhibitor, SNPBIX, that inhibits hemoxygenase 1, and we infected the flies, and then we gave this inhibitor just the day before transmission, at the day of the transmission, and the day after, to just block this the effect of hemoxygenase during that time. And then again, we saw the same exacerbation of the pathology without any really obvious effect on the parasite load. And this probably is occurring because hemoxygenase, as I told you, has byproducts and has been really shown in many studies, some of which are listed here, where the more hemoxygenase there is, the less inflammation there is. It's an potent anti-inflammatory through its byproduct after the hemoxygenase 1 reaction. And then to look at that, we again did a targeted gene expression array. And what we could see here, and so this is just to show you the whole array, but what was significant is when we remove the hemoxygenase from the equation, we do see an increase in inflammation through increase in IL-1 beta, a significant increase in CXCL1, CCCL2, and CHIL3, which is a collagenase. And also we see ferroportin, which is an iron transporter, and HEMOX1, which is the gene for induction of hemoxygenase, also increasing potentially because the cells are not able to break down the RBCs. So there is a clear effect when we inhibit the hemoxygenase on the I mean, this is the take-home message on the micro-inflammatory environment at the bite site. So remember this from the earlier in the talk where I showed you that the IL-1 beta actually comes under natural conditions after the bite, around 6 hours, with a very potent production through neutrophils. So we decided to look in another, and this was done in Lutzumia longipalpus infected with Donovani. So we decided to look at L major infected with Dibowski because that's where we looked at the disease pathology since it was easier. And all you need to see from here is, again, at 6 hours, we really see a population that's strong, that's producing IL-1 beta. So it shows you that this response is universal in a different parasite vector pair, and that it's mostly coming 70% from neutrophils. So at this time point, what would happen, trying to mimic what is the effect of hemoxygenase later on? Because we know this is a loop that intensifies the infiltration. So if we inject COM2, which is a carbon monoxide donor, and if you remember, carbon monoxide is one of the anti-inflammatory byproduct of hemoxygenase reaction. So what we did is we infected sandflies, transmitted to the mouse, 2 hours later injected COM2 into the skin, and 4 hours later did the flow cytometry, which is 6 hours after the bite. And what we can see here is that there's a definite significant reduction of IL-1 beta producing neutrophils, and also a reduction in the number of neutrophils coming in, although it was not really significant. So in this summary, we showed the other side of the coin, that in fact the host immune response is vital also for establishing the parasite. We showed that hemoxygenase is induced after the bite. We identified a new resident subset of professional iron recycling macrophages in the skin that are mopping up these leaked RBCs, and that this has an effect on disease, because in the absence of hemoxygenase, we see increased inflammation, exacerbated pathology that suggests that hemoxygenase-mediated control of tissue damage at the bite site of Leishmania-affected sandflies does promote tolerance to leishmaniasis, which is probably important so that the parasites are tolerated. And if I have a little bit of time, I want to continue with another really cool project that we did in the lab, because what I showed you right now is the sandfly feeds, gets infected, it develops a mature infection, it transmits. And that has huge consequences on the host immune response because of all the things it actually instigates or starts through the damage it causes to the capillaries, but also due to the components that it injects. But a very talented postdoc that's now a research fellow in our group, Tiago Serafim, asked a simple question. So that's all great, but we know only a few parasites are ingested into the host. So what happens to the Leishmania that remains in the gut? And that produced a phenomenal set of studies that are actually published in Nature Microbiology in 2018. So just to remind you quickly the life cycle, when the fly takes a blood meal, it takes up amastigotes, they develop into their cycle, produces metacyclics, and we thought that was the end of it because this is the last developmental stage. It's terminally differentiated, it's just at the mouth parts, there's nowhere to go except to be transmitted to the host. Well, very wrong. So if we ask the question, so what's happening inside the fly? So what Tiago did is what we always do, infect the flies, count the parasites per midgut in the upper panel, and look at the percent metacyclics as they develop. And when we give a blood meal, the parasite load increases, the metacyclics begin to appear and increase in number. But what Tiago found is if you have a mature infection and you give another blood meal. So imagine this is the parasite that is transmitting this is the fly that's transmitting the parasite and causing all the things I showed you before. But actually when the fly feeds and takes another blood meal, here in orange compared to the blue, something really remarkable happens. The metacyclics that were extremely high at day 12 in blue and orange, once we give a fresh blood meal, we were astonished that they just disappeared. The metacyclics disappeared. So you can see here that they go down for another 2 to 3 days. And then they emerge again, and they're much more coherent as a population. Not only that, if you look and compare the percent metacyclics to the parasite load, you can see that and this is a percentage of the parasite load, that it is much more in number, because the number of parasites has also increased compared to one that only took a single blood meal. So wow, that means that every time an infected fly feeds, it actually makes its infection better, makes it have more metacyclics that are able to infect. And the second subsequent additional blood meal is not infected, could come from chicken, could come from humans, could come from any animal, and you will see the same very striking phenomena. And here, I don't know if you can see the videos, so I'll start both of them. So what we see when we actually cut the anterior end and look 2 days after we give half the flies another blood meal, the video here on this side, is it playing? Anyway, you can see these parasites are larger, move more slowly than these parasites, which don't want to play. It played at the beginning, that are pure metacyclics and moving like crazy. And when Tiago looked by electron microscopy, you can see that this is what he finds when we don't give another blood meal. But 2 days after we give the blood meal, there is almost no metacyclics. What we see is this new life stage that we actually call the retroleptomonad promastigote. And just he captured in a very elegant and simple assay by putting metacyclics on polylysine slides and simply putting them in a serum environment with all the components from the serum. We know it's a serum component. You can actually physically see the resorbent of the long flagellum of the metacyclic and the induction and differentiation into the retroleptomonad from the metacyclic. It's pretty cool. This is another one. I'll quickly show it. I'm almost done. But I thought this would be interesting to this audience to realize that sandflies, with every blood meal, if they're infected, can actually amplify their infection. So it's not all infected sandflies are equal, and that may be what reflects upon all the divergent pathologies we see in the people. So just to show you that this is even more crucial than you would think, because the experiments that I showed you before is how we infect sandflies classically through membrane feeding. We put a certain number of parasites that would come out to, I don't know, 500 per sandflies or something like this. But in reality, what they pick up from a lesion might be much less. So what I want to show you here in one of the final slides is that if we take Lutzomyia longipalpus and Leishmania infantum, and we actually get them to feed on a sick hamster, you can see that in blue, when they only take a blood meal, the number of parasite and the percent metacyclics are really very low, because it's a struggle to develop the infection from the few parasites they picked up from the animal. But if we give them a second blood meal, then we recover the infection, and the infection progresses and produces nice metacyclics. And this is also the same is observed with Phlebotomus papatazi and Leishmania major, a completely different vector parasite pair that transmits cutaneous leishmaniasis. And the implications of that, and he did single fly transmission, so one single fly from each group, one that took an additional blood meal or one that didn't. You can see that this is the percentage of mice that developed lesion after the bites of these flies. And there's a significant enhancement of the success of transmission to the host when the sandfly has taken more blood. And so this is really, I think, a big story that will really need several more years for us to fully explore, that shows how really even the multiple blood meal that the fly is taking to lay eggs, nothing to do with the infection, has a tremendous implication for the parasites, for transmission to the host, and for disease pathology. And more, the last slide that is really unpublished yet, it's not... we're currently analyzing it, that was done by Iliano, actually shows that these retroleptomonad, and this is RNA-seq from parasite residing in the gut at different time points, and the retroleptomonads really seem to be separating from any of the other stages into their own bona fide stage. So basically, what I showed you in the last part, this is the summary, is just by simply asking the question, what's happening to the parasite after it takes the blood? We could discover a really microcycle that produced a different new stage, the retroleptomonad, that is changing the equation for the ability of infected sandflies to transmit. So this is our group at the VMBS at LMVR. I don't know if you can see Jesus here, and this is Tiago, as you saw before, Iliano and Tiago, Eva. Renedy was not with us, and I have to thank Claudio too, because he is the colony manager, and without his care of these colonies that produce thousands of flies for us to manipulate and infect, and he really wants Phlebotomus argentipes as well, we would not be able to do this work. Miguel Soares gave us the hemoxygenase mice with lots of discussion. Valeria Borges really gave me the idea to look at hemoxygenase 1 from Fiocruz, Brazil. Here are Renedier, are part of all the projects I presented. We interact a lot with them. And the exosome study, as I said before, was done with Martin Olivier and Vanessa from McGill University, with a past lab member who's now in France. So thank you very much for listening.
Thank you. Thanks a lot, Sharon. What an illuminating lecture. I think there are lots of questions in the chat box. So I would like to hand over the session to Dr. Devarati Mukherjee, who has recently awarded PhD from our laboratory, just to take over the charge of question and answer session. Please, Dr. Devarati Mukherjee.
Am I audible, sir?
Yeah, you are audible. Please go ahead.
Yeah. Good evening, everybody. Thanks a lot, ma'am, for such a wonderful presentation. So I can see lots of questions in the chat box. First question is, do the sandfly gut microbiota affect the survivability of neutrophils?
No, actually, we have not looked. I mean, David Sachs has shown that the neutrophils become partly apoptotic at the bite site, and that's why the macrophages don't kill them. But we have not seen any effect, direct effect of the microbiota on the neutrophils. In fact, they look healthy and produce a lot of IL-1 beta.
Okay, ma'am. The next question is, what is the antibiotic cocktail used?
Yeah, that's a good question. So we actually really tested a lot of cocktails, and a lot of them harmed the parasites. So we had to be very careful. And we ended up with a combination of penicillin, gentamicin, and clindamycin. And the combination and their concentration is present in the paper, the details. And these were first tested on cultures, and then we tested them inside the fly on the infection. And regarding their effect, and that's also a very good question. So when we were looking at removing the microbiota from the fly without affecting the infection, because remember I told you we did a previous study that's actually published in MBio, if you look it up, where we show in the absence of microbiota, the parasites will not develop. Microbiota is an essential component of the healthy development of the parasite in the gut. But in this case, for our purpose, we allowed the infection to mature completely, that it was ready to transmit with lots of metacyclics, and only gave the antibiotic cocktail for 36 hours before we transmitted. And we showed that the parasites are healthy, able to infect cells in vitro. The flies feed well. There's no effect on mortality. So this kind of transient antibiotic treatment worked very well for this purpose.
The next question is there any dependence on gram character of gut microbiota on the recruitment of the lymphocytes?
Yeah, so these animals are innate, right? So, I mean, they're naive.
Yes.
They've got no adaptive response to the Leishmania or anything else. So we don't see a lot of lymphocytes in these early events, but it's actually a very good question and something we want to explore. We are exploring, well, before the COVID-19 pause, of course, is that, in fact, this IL-1 beta induction might have a huge role in developing this very strong adaptive immunity to Leishmania that people say are coming from a natural bite from the Leishmanization, because IL-1 beta, we know, is quite important for the promotion of a Th1 helper lymphocyte helper profile. So we have not really explored that, because this is a window where really we were looking at the innate events in a naive host, and now that is the next phase. Obviously the next question is, how does that affect the development of adaptive immunity and lymphocytes? Good question.
The next question is, does sandfly bite produce hypersensitivity type 1 reaction like those of hypersensitivity in mosquito bite?
Actually, and Jesus will talk a lot more about that, because you do develop delayed hypersensitivity after sandfly bite that is quite strong and lasts for years until you know, almost when you're in the 40s or 30s, you begin to get them, because we did a study in the field in Mali, and children have DTH to sandflies at age 2 and actually retain it till they're more than 40. While in mosquitoes, then this actually disappears much faster and you get immediate and then you get desensitized. That's not the case for sandflies, but a DTH response is an adaptive response to the saliva. So you need the mice that have been bitten several times, which is something I didn't touch on in this lecture.
And the next question is, drawing from a question asked to Professor Nakashi, would HO1 be responsible for the cause of anemia in Leishmaniasis patients?
No, I don't think so. That might be related to the disruption in the structure of the spleen, actually, I would think, which is very important to recycle red blood cells. And the iron that is recycled goes back to the bone marrow to make more RBCs. And so if you're destroying the spleen, then you're destroying the capacity of these cells through hemoxygenase, but in the spleen, not in the skin, to actually recycle the iron that is needed to make more RBCs. And that's potentially, I mean, I'm not a clinician either, but this is how I would think it would affect the anemia that we see.
If I could respond to that question, this is here. So, in next few minutes. So, I just found a paper by Dr. Abhay Satowska, which I sent it to Chiranjib, that
Okay, sir.
what it does is that the Leishmania donawani with infection induces the erythropoiesis, and that anemia is associated with increased level of erythropoietin and increases the other transcription factors and downregulates the alpha globin, beta globin expression, mRNA, and their transcription factors. And that could be a mechanism by which the Leishmania donawani causes the anemia. Thank you.
Anemia. Thanks a lot, sir. Moving on to the next question. Did the activation of NLRP3 inflammasome by gut microbiota lead to the activation of IL-1, IL-18 and the inflammatory process downstream to that, or is it solely IL-1 beta dependent?
No. We are certain it actually involves IL-18 as well. The problem with IL-18 is that it's constitutively produced in the cytoplasm. And so it's very hard to detect whether it's being produced or upregulated. We looked at it, and in our time points, which are very early, so many studies have looked at it 7 days later, 2 weeks later, but that early on, it's very difficult to distinguish the IL-18 that is still in the cytoplasm from that is actually being released. So we wouldn't know if it's actually, we wouldn't be able to prove it's actually being secreted. What with the IL-1 beta, the nice thing is that it gets cleaved and then it becomes active and secreted. First of all, the IL-1 beta is inducible, so you need the NF-kappa beta to start to transcribe it along with the NLRP3, and we could prove that that is what's occurring. We looked at IL-18 at this time, but it was hard to say. But it makes sense that it is because they go hand in hand.
Okay. Does microbe-dependent inflammasome activation and IL-1 beta production are sufficient for parasite dissemination, or it also needs other cofactors in the infectious inoculation to initiate the inflammatory response?
Yes, so that's a very good question, and that's what I've actually been saying, is that the events that we focused on was on the IL-1 beta, but in fact, we do know that many other factors do other things that are equally important. For example, Paul Bates and Matt Rogers showed that the promasticot secretory gel actually switches the macrophages at the site into an M2 phenotype by switching the pathway to the arginase metabolism. He showed it in a very elegant way. So that's also happening. So that's a good question, and that's what I want the audience to imagine, in fact, that it is the microbiota, the promasticot secretory gel. Saliva has many very, very important, because apart from the immunogenicity and immunomodulatory functions of the protein, even the physiological aspects of their role, which is vasodilation, inhibiting coagulation so the host bleeds more, is probably why the bite is so efficient at producing hemoxygenase and causing these RBC leakages. It's very different from the needle poking, as I hope you saw, where, the hemostasis is restored pretty quickly because there's nothing to prevent it. So correct. I'm not saying microbiota is the only thing, but it's a really major initial step, because the triggering of the inflammasome is almost instantaneous when there is toll-like receptor activation, TLR-L2, by the microbiota, and then everything comes after. But all of them are producing something, and that's why at the end I told you to imagine a very potent, very hyper-inflammatory milieu at the bite site from all these components.
The next question is, in which context a 6-day infection interval for 2 times are followed before transmission?
I'm sorry, it's not clear, the voice. Can you please?
Okay, yeah, of course, ma'am. In which context a 6-day infection interval for 2 times are followed before transmission?
Oh, 6-day interval, you said?
Yes,
I'm
ma'am.
sorry,
6-day infection interval.
Okay, so I'm sorry, I still don't understand the question, but I know I'll try to answer something about 6 days interval. It's so the sandfly actually feeds every 5 to 6 days. So you're correct. And I gave you a flavor of the importance of subsequent blood meals when the metacyclics are there in a mature infection, because it really amplifies the capacity to transmit. But in fact, the second blood meal that an infected fly would take would come at 6 days. Is that your question?
Yes, ma'am.
And if that is the case, then it actually does the same thing. It sets the parasites back to cause them to multiply much faster and produce a much stronger homogeneous infection for the development of the metacyclics. I didn't show that data though.
Okay. Moving on to the next question. During an infected bite, whether promasticot secretory gel could help in the survival of gut-digested microbiota in host?
I don't think so because, yeah, I mean, in fact, to me, the promastigot secretory gel, which is immunomodulatory, we know its constituent, but in the act of transmission, it is crucial because it is actually so sticky. It's made of sugars, soluble sugars. So it sits right at the anterior of the gut behind the mouth parts. And that's why transmitting all these other components is so efficient in sandflies. So the parasites are stuck in the gel, the bacteria, the exosomes, and they are all transmitted in this gel that is soluble. And then they find the cells. So does it protect it? I don't know. I have no answer. But I think the immune system is so refined to detect bacteria and quickly respond to it with these inflammasomes that the bacteria is gone very quickly after being injected into the host. And we, in fact, don't see abscesses or anything developing at the sites of the bite.
Okay, thanks a lot, ma'am. There's no more questions left. So
Great.
thank you for the presentation. I will hand over the session to Dr. Pal. Sir.
Thanks a lot, Sharon. Thanks a lot. I would like to convey my gratitude and thankfulness on behalf of West Bengal State University. Thanks a lot for your presence in our session, and we have enjoyed a lot and learned a lot regarding the sandfly vector and the host interaction, the complex immunology behind that. Thanks a lot. Thanks a lot, Sharon.
Thank you. It was a pleasure.
Thank you. Thank you. Yeah. So the next session, I would like to introduce Dr. Jesus Valenzuela. Dr. Valenzuela received his PhD in biochemistry from the University of Arizona in 1995. He joined the Laboratory of Malaria and Vector Research, National Institute of Health, USA in October 2002. Dr. Valenzuela became a senior investigator in October 2009. At present, he is the chief of Vector Molecular Biology section at NIH. The key focus of his research is the development of robust protein recombinant expression and purification methodology to test salivary proteins for biological activities and validate vaccine candidates identifying using DNA immunization. Professor Valenzuela is the key scientist involved in the recent vaccine trial of the university mosquito vaccine in humans by NIH. He has published more than 180 articles in his credit. Today, he will discuss and share his experience on basic and translational research of vector salivary proteins. Dr. Valenzuela, sir, please.
Well, first, Chiranjib, thank you very much for the invitation. Claudio sent his regards from the Sandfly colony. I remember you and your family still when you were here.
Yes.
It was a great time when you visit our lab, and I had a great stay here in the lab. I'm glad to reconnect. Today I'll present some of the work we're working regarding to vaccines. I think Sharon needs to stop presenting.
Stop presenting.
For me to be able to get the... Sharon, I think you need to stop presenting. Let me see. Yes.
Yeah.
Sorry.
All right. Share your entire screen. Allow. Let's see.
Perfect.
Let me see now. Okay. Can you see? Can you use the mouse?
Yeah.
Or not?
Yeah, you can use the mouse.
Okay. And let me see if there's another way to do it too. Can you see this? The laser.
Yeah.
Okay, good. All right. Again, thank you a lot for the invitation. Today, we'll talk about our efforts to move a lot of our findings in animal models into humans. We still continue doing a lot of the animal models, but I think one of our objectives is to translate as much information as possible to clinical trials. Then, just to start this this session, well, also, if you like to interrupt me in any during the talk, I'll be happy to take any questions in the middle. Or if you want to wait, that's fine. But either way is fine. I know that it's late. It's about like 7 something there. Then I'll be brief, and but I'll be happy to discuss anything. Well, I'll just start with what Sharon mentioned before, that the importance of the infectious inoculum, that still we have this knowledge gap in vector-borne diseases, not only in sandflies and leishmaniasis, also in viruses, mosquitoes, malaria, plasmodium, ticks, and bacteria. The concept of what Sharon talked about, of the injection of the pathogens together with saliva, exosomes, is still very new. And we need to study that. And that's what I'm trying to convey today. How are we moving from what we're learning in the animal models to what we learn from Sharon's work to go more into humans? And this is a figure that I hope it goes now to textbooks. We normally know that, or in textbooks, they show just parasite going into neutrophils and macrophages and their detection, but we all know now, and there is extensive literature, that insects, in this case sandflies, deliver parasites. And together with parasites, and as Sharon showed, they deliver salivary proteins, PSG, exosomes, and now we know more about digested microbiota. And all this has tremendous impact and influence in disease transmission. In terms of saliva, we know that those salivary components have very potent activities, pharmacological activities, like inhibiting the contact pathway activation, inhibiting complement. Some of them are actually endonucleases that can destroy extracellular traps. And we also found potent anticoagulant that has dual function. Not only they inhibit coagulation, but also they can inhibit the alternate pathway of complement. Then saliva is quite strong. Somebody asked a question that if the microbiota alone or any component alone will do this. And I think what we're learning now is that it's the system is very redundant. We are seeing a complementary activities, some activities that are found in the host, some activities that are delivered by the saliva, for example, anticomplements, anticoagulants. We have anticoagulants as human, as mammals, and also parasites, they have other components. Then what we're seeing is the infected inoculum is molecules that are complementary, that are very redundant. And that's a very important concept to remember. Remember that Karen showed you that the microbiota activates IL-1 beta, and that brings neutrophils. Well, saliva has a complementary activity. We found something very interesting and more recent findings. We are the paper is in the review, but I'd like to show you some of the data. Shani mentioned that when you have an either infected or uninfected sandfly bite, you have lots of neutrophils that are recruited. This was shown by Nathan Peters and David Sachs. But we saw that also this that it was very different from you. Then sandfly should have something to do. Then already Sharon showed that microbiota goes to the IL-1 beta, but we also knew that could be another mechanism. Then we asked the question, can saliva also attract neutrophil independent of IL-1 beta? Okay, just to remember this. And the answer is actually yes. If you use, this is a neutrophil recruitment or chemotaxis assay. If you add just buffer or some control, there is just normal, no recruitment. I mean, the neutrophils has to travel from here to here. There's nothing. But if you put saliva of sandflies, something different happens. Oops. Oh, I see. You know why? Because I have the point. Okay. Then you see something very different. The neutrophils start moving into where the saliva is. Then that's already telling us that saliva has a chemoattracting activity. Then we need to identify what factor actually is in saliva. Then we tried different proteins, and we found out that there is a family that is doing this, that is called the yellow-related proteins, which is, I'll show you more of the structure. But when we inject either PBS or the recombinant yellow proteins or whole saliva, we see here a tremendous neutrophil recruitment in comparison to only PBS injection. And you will see here, and this is on a space of few hours. You will see a tremendous, and this is in vivo imaging, of neutrophils being recruited in the skin. Then saliva is quite powerful, actually, or these salivary proteins are very powerful to bring these neutrophils to the site. We found actually that this neutrophil chemoattractant, although similar to the activity of chemokines, they are not chemokines. The structure has been solved by John Anderson and in the department, and it shows that the structure of this yellow family of protein, the sandfly neutrophil chemoattractant, is very different to the chemokines, completely different. This is a molecule of about 7 to 12 kilodaltons when it's actually in that's a dimer. This molecule of the saliva is about 43 kilodalton as a monomer. We're finding that actually, I'm not going to show the data, but this is working through a G-coupled receptor. But it's going to be very different to the CXCL receptor. Then here we have now something unique. We have a molecule from an insect that is attracting a mammalian cell, a neutrophil. This is the first account of a molecule that can do something like this from a isolated and identified from an arthropod. We show also that when you co-inject these proteins, either saliva, this is when you inject Leishmania major only in animals, they develop some level of infection. When you add Leishmania plus saliva, yes, there is more infection. When you add saliva plus these yellow proteins, there is also a lot of infection, showing that this protein actually helps the parasite to get established. When you add other recombinant proteins, that is nothing related to the yellow protein, there is no increase in infection. Then what we're seeing here is another way that saliva is actually potentiating Leishmania. It's just helping the parasite to move into the neutrophil And actually, that's what we're seeing, more neutrophil infected with Leishmania. And then this neutrophil is going to go later to the macrophage and cause an infection. Then, just to summarize this part, we're and to connect to what Sharon showed, is we are seeing this tremendous effect of what we call vector-derived factors in the skin of animals that are affecting the development of pathogenesis of some vector-borne diseases. In this case, we show leishmaniasis, but we're seeing, other people have seen also with viruses and mosquitoes, just to give you an example. Now, we found actually many years ago, and we know that saliva promotes infection in animals, but we also learned a few years ago that if animals develop an immune response to eat insect saliva, they are protected from infection. And one of the first labs to do that, we collaborated with David Sachs at that time, and one of the first persons to show that was actually Sharon in a science paper where she showed that immunity to whole bites can protect animals against infection. Then we decided to investigate what are these molecules, and then the last decade, I would say, is investigating and trying to identify these molecules. Then, and the mechanism. We found few of these proteins. I'm going to show you an example, and this. But just to give you the landmark of protection of saliva for sandflies and leishmaniasis, either an infected sandfly bites, what Sharon showed many years ago, and we were able to reproduce this with sandfly recombinant proteins, is that they produce a CD4 T cell response, a classical delay type hypersensitivity response that brings a lot of T cells. And it's actually the environment is a Th1. It's more interferon gamma than other Th2 molecules. Then this is the landmark of protection. Somebody asked if insects or sandflies will generate a type 1 hypersensitivity response, and they do. But actually what we're looking for are not those molecules. We're looking for molecules that are producing more beneficial and less aggressive response, which is a delay type of hypersensitivity with this feature, CD4 T cells and interferon gamma. And we have shown if animals have this landmark, they protect against cutaneous leishmaniasis or they protect against visceral leishmaniasis. So this is in the rodent model. When we move into rhesus macaques, we use a DNA vaccine in combination with recombinant protein. We inject rhesus macaques and challenge them with infected sandflies with Leishmania major. And we showed that a tremendous protection in the group that was vaccinated with SP15 proteins. This is the structure of this is a unique protein in sandflies. It's called PDSP15. It's a 15 kilodaltons odorant binding protein like. This is very important. There were 3 monkeys that did not produce an immune response to this vaccination, and those animals were not protected. Then that's a very good correlate of protection. All the animals that had an activity at Th1, interferon gamma were protected versus those 3 that were not. And we see a tremendous difference in the lesions. Well, the control has very open lesions. This one had or very little or nothing. And this is these were showing the worst. There's some level of lesions, but many of the monkeys did not have any lesions. But to us, we found something extremely important in this animal model, is when you stimulate PBMCs from these animals with Leishmania antigens, we see that the animals that were vaccinated with SP15 produced lots of CD4 interferon gamma to Leishmania, but not control. What this is telling us is that the sandfly saliva immunity is enhancing anti-Leishmania immunity. It's creating an environment that is going to help Leishmania immunity. And that for us was quite interesting. And that's one of the concepts that we're following now, that saliva may not be working as a only vaccine. I think we may need to combine salivary protein with the pathogen. and I'll show you some other ideas about it. This is the concept of saliva vaccine, how we think it works. We think that immunization with a salivary protein will, the protein will be taken by antigen-presenting cells, showing to T cells. T cells will be primed and ready in a Th1 environment. When the stem fly delivers the parasite together with the salivary proteins, this is going to be again taken by antigen-presenting cell, but now the presentation is going to be with the memory, with cells, T cells that have the memory against the salivary antigens. And then these cells are going to start releasing cytokines that will activate macrophages that will kill the parasites. Also, this environment right here not only kills parasites, but this environment now, which is Th1, the cells will see the parasite with a Th1 environment too. That's why we hypothesize how saliva is increasing anti-Leishmanian immunity, because the immunity to the saliva is promoting an environment that will have the antigens of Leishmania presented as a Th1 as well. That's the concept. That's what we're looking for. Now, how do we move from animal models to humans? Then we initiated with 2 studies, and I'm going to show you the other current 2 that we did. The first one was to demonstrate that saliva actually, or bites in humans will produce a Th1. We needed to demonstrate that. We knew that in animal models, we could see this Th1, and the Th1 was protective. Then the first question was, can we find a saliva-specific immunity Th1 in individuals that are naturally exposed to sandflies? And we went to Mali to perform this study. The second study, what we'll call bench to the clinic, is the identification of Th1 inducing salivary proteins in humans, and this is more or less a reverse immunology approach that I'm going to show you. Then we have these 2 initial clinical studies. The first study, I'm not going to spend much time here. I prefer to discuss if you have any question. People were exposed. Then we get these individuals and expose them to uninfected sandflies that were colonized. We took skin biopsies and asked the question, are these individuals producing a cellular immune response? And the answer was yes, they produce DTH actually. And the next question was, what type of DTH are these individuals producing? And it was a Th1 type response. This figure we published a few years ago showed that the individuals that were producing a DTH to a sandfly bite, this environment was a Th1 type. Was a lot of interferon gamma, less of other Th2 cytokines. Then we concluded that if we can translate this concept in the animal models to find a vaccine or a component of a vaccine in humans, this will likely work because humans are producing a Th1 immune responses to the salivary proteins injected by the sandfly bites. Then the next question was, can we identify molecules in the saliva? Can we identify the molecules in saliva of sandflies that are producing Th1 immune responses in humans? That was a bit challenging. And what we did actually was to expose people, and it was in collaboration with the Walter Reed Research Army Institute with Naomi Aronson. And the experimental design was not that complicated. It was more complicated to follow. It was actually to expose people to sandflies for some time. But then we did this many times and followed them for almost a year and see if they would produce an immune response. And the answer is yes, they produced a typical DTH response, and we were confident there was a Th1. Now, what molecules are producing this Th1 response in humans? And to do this, we did a reverse immunology type approach. Then we identified the most abundant salivary proteins from the flies, in this case, Lutzomyelon palpis, and we can optimize the sequences, the transcripts, and put them in a high-throughput vector, DNA vector, in which you can transfect cells, purify the protein to a single molecule, or almost 99%. And also we show endotoxin to be very, very low. Anything about I remember we tested 0.03 EU per ml will activate cells. Then that will be a no-no. Then we needed to show that the activity we were seeing in cells was actually coming from the protein and not from LPS or any endotoxin coming from the preparation. Then we excluded some proteins. Anything that was with the cutoff, below the cutoff, we use it. Then we got the PBMCs of these individuals that were highly exposed, and we tested all these molecules, and we tested many times. And what I'm going to show you is one slide that shows the summary of many individuals, many trials with all these different proteins. Then what we're seeing here is actually the activation of PBMCs and the measurement of interferon gamma. Then, and what you see is the levels of interferon gamma in those individuals. And we saw some proteins that are active, and actually these two and these two are the same. This one is different, I will call the number 3. But we saw these two that were very consistent, very high. LGL143, which is the anticoagulant, and LGL19, which is actually an anticoagulant. It's a very small protein. This is about 38 kilodalton proteins, and this is a 19 kilodalton proteins. Then we have now, actually, through this reverse immunology approach, 2 very good candidates to humans. But first, we needed to show that any of these 2 could protect animals, although we can go humans, but we wanted to further prove this. Then, in collaboration with Hira's lab, particularly Ranadir, we show that immunization, or Ranadir showed, that if he immunizes mice with only I think it was 1 or 2 micrograms of recombinant protein with no adjuvant. That's very impressive for a vaccine. And then challenge these animals with infected flies. All the animals that were vaccinated with LGM19 survived. None of the animals, or most of the animals with the control vaccine, PBS or BSA, they did not survive. Also, there's way a larger parasite load in the control versus LGM19, showing there is a very good vaccine, and also the size of the spleen. This molecule is a very tiny molecule of 11 kilodalton. It's very new, only found in sandflies, and actually in sandflies from the family of anticomplements in the Lutzomyia. But we found something, and I'm not going to show the data, but we found something very, very interesting. The 2 molecules that we have seen to work very well in animal models or humans by PBMCs are 2, the SP15 and the LGM19. If we overlap the structure of LGM19 and SP15, we found a domain of other binding protein. And we think that domain is the one that is activating the immune system to produce this Th1 environment. We need to do those tests, but that to us was a new finding, and only based on the structure and only based on the ability of these molecules to protect animals against leishmaniasis, cutaneous and visceral. Then again, this molecule is very strong in protecting this. Now, these are molecules that we like to move forward for vaccine in humans. In the meantime, we were contacted by the NIH and by a company called SICK to see if we were interested in participating in a study. And the study was to test, actually, a saliva peptide vaccine, which is follow the same concept we're doing with some flies. And over the years, there has been a lot of resistance for the concept of a saliva vaccine, because the idea or the fear was that it would produce an allergic and really bad response in humans, that should not be even considered. Then we have a tremendous opportunity here to test if a vaccine coming from a mosquito saliva protein will be safe. And that, if so, this will open the door to other vector-borne disease vaccines based on saliva. Then For this, we teamed up with the LID, Laboratory of Infectious Diseases, and particularly the clinical study unit with Dr. Matt Memoli. He's a physician. And at that time was Jessica Manning, which is actually the first author of the Lancet paper. That this was the result of the Lancet paper was the result of this work. We have the LMVR, of course, and many other NSIC, the collaborators that provide us this peptide, and they did this peptide vaccine based on their own algorithm to identify peptides from the saliva of mosquitoes. And they show already for flu studies and other studies that their approach worked very well. Then they have the patent, they have the GMP production of the molecule, they have all the safety studies. Then what we needed to do was to move this to humans. It was a golden opportunity for me, for Sharon, Fabiano, for all of us that were working in the LMVR and that have worked with saliva for many years. Vaccine concept. Then what we did was this, and we participated in actually not only in just the concept, but actually we helped in writing the protocol, going with the assays, human studies. It was in my view a beautiful experience of something that we for many years wanted to do, is to move to human trials. Then this was a randomized, double-blind, placebo-controlled, single-center phase 1 study, with the main objective to show for safety and, of course, immunogenicity. But the most important part was safety. We wanted to show that saliva-based vaccine was safe. Then we enrolled volunteers, 45 volunteers, more than 45. 45 were the goal, but were about 49. And there were 3 cohorts. The first cohort was just the peptide vaccine only, that was 15 individuals. The second was the peptide with the adjuvant montadine, and the third group was placebo, just water. And the vaccine was done subcutaneously. To make the story short, we saw the individuals actually produce antibodies against the salivary peptides, very, very good level of antibodies. Again, 102 days follow-up, time. In comparison to the placebo group, we observed this only in the adjuvanted vaccine group. We did not see in the vaccine-only group the IgG response. However, the cellular response we saw in both groups. We saw the vaccine group and the placebo group have significant levels, not a lot, but significant levels to the placebo group in producing interferon gamma and other cytokines. But to us, we wanted to demonstrate that, yes, these molecules can induce an immune response, antibodies, and also cellular immune response. Yes, they did. But the most important, remember, was the safety. Then, this is the conclusion at the top. We did not observe severe adverse event. There was one participant that with the adjuvanted vaccine that has grade 3 injection site rash. But other than that, we did not see any anaphylaxis that people were afraid of. We didn't see any major problem with the vaccine. Then we were quite excited about the results. I think the immunogenicity can be improved. Remember, this is the first trial ever for a saliva vaccine for many years. And I mentioned here, this is the first study in which humans have been vaccinated with vector saliva of any kind. Then to us was a breakthrough, was the ability to demonstrate that this concept can be used in humans. Then I read the last one, is this finding suggests that vector-targeted vaccine administration in humans is safe and could be a viable option for the increasing burden of vector-borne diseases. Because this is not only for mosquitoes, this is for some flies, this is for viruses transmitted by mosquitoes, this is for malaria transmitted by mosquitoes, this is for ticks, for other diseases transmitted by ticks, any other vector-borne diseases. This is open a new door. That's why we are quite excited with these results. Now, I'm going to show another clinical study that we are participating. Which is this? This is a very important concept. This was actually in a meeting that we realized this. We were talking about vaccines, and we realized that most phase 1 or phase 2 clinical trials are mostly done with normal healthy individuals. And I'm talking about vector-borne diseases, okay? I'm talking about malaria, dengue, leishmania, any other thing, vector-borne disease you can think of. But then think about this. When you're deploying a vaccine into the field, we're talking about phase 3 in this case, we have to remember that a great proportion of individuals will be exposed to insect bites. Actually, some flies, for example, in India, or even mosquitoes in India, and other blood-feeding insects. And this is not only from India, Africa, America, South America, and that will be very common. Then our question goes, how do humans respond in the skin to multiple insect exposure? And more important, could this skin response to insect bite affect vaccine efficacy? We have seen already many trials in which the beautiful vaccine studies, phase 1, showing everything, but when they move to the field, they fail. And that happens to malaria, or they don't work as thought they would work. Happened to leishmania, happened to other phases, other studies. And is it possible that there is something that we need to learn and to get more knowledge in that area? And I think that's this area of the skin. Then we decided to do a new study, which is called we call it the BITE study, which is to understand the early skin immune responses to insect bites in healthy human volunteers. And this is insect bites in general. We wanted, and actually we took three important vectors. Yes? Oh, can you hear me? Good. Oh, can you see it? Is that okay? Can you hear?
Jesus, it was okay, but now it's...
No, because I heard something. No, I heard something, and I thought that there was a question or something went wrong. Okay, then can you hear me now? Yes, good. Then I was saying that why insect bites? Because I think we wanted to understand vectors in general, not only sandflies. And although our main insect in the lab is Lusomia longipopis and other sandflies, phlebotomus, we wanted to understand also Aedes aegypti for dengue, chikungunya, Zika, Anopheles gambiae for malaria. And actually, we're doing another study. We submitted a proposal for ticks. And the idea was to do a study about the skin of this after a bite. Then how do we design this study? Then we wanted to know two things. We wanted to know what happens after a single insect bite, either with Aedes, Anopheles, or Lusomia. Then we have the mosquito bite a human. Then we take 30 minutes skin biopsy or 4 hours skin biopsy. Why do we want to do this? We want to understand the early innate response. And just to give you a background of why the study started, actually was after Chiranjib published the paper of the microbiota. We were in the hallway and I said, Chiranjib, will it be the same in humans? And we couldn't find anything. Then we decided, Chiranjib and I and Fabiano, and met with Matt Memel again to move this forward and to ask the same question that Chiranjib asked the mouse model. And we do it in humans, and we went ahead and do this. And the same thing is we wanted to understand what is the early innate immune response in these humans, in these volunteers. But also remember, we wanted to, in terms of vaccine development or other things we want to know, is most of individuals in the endemic areas will be exposed to insect bites. Then what we did was what is called an experience arm, in which we expose individuals to bites. I think it was 10 or 20 bites every 2 weeks until they develop an immune response. Then we again take a biopsy 4 hours after the bite, for bite number 4, and 48 hours again to see the innate response, although we should some level of adaptive here too, but also focus on the adaptive response at 48 hours. Then it's a good number because it's 15 subjects per insect. And this is our feeding protocol. All our insects are going to be contained here with this will have a mesh, and the mosquito or the sandfly will bite the individual, and then the individual we will score, and then we will take biopsies. Then this is a scoring of the individual. You can see for Aedes aegypti, very tiny immune responses when the individual doesn't have, is naive or not much encounter with the mosquito. These black dots represent actually surgical markers because this position the biopsy site. Then when we're seeing insects, we always believe that insects are the same, but they're not. And we could see already a significant difference between Lutzomyia longipalpis and Aedes aegypti, and this individual has never seen Lutzomyia longipalpis before. And this actually is an erythema, and it's not inflammation. It's actually induced by a molecule in the saliva of sandflies called maxadillan. It's one of the most potent vasodilator known in nature. That's very potent vasodilator. Then we start seeing in humans already difference between different vectors. Now, what happens when you compare early time points to late time points in the skin when we do biopsies of these sites? And this is what we see. And the unbeaten site is very quiet. At 4 hours, this is experience. This is an individual that has been beaten many times. And we start seeing lots of cells. At 48 hours, we also continue seeing lots of cells. And very similar to the cell recruitment we saw in the animal model, we're seeing it also in humans, a textbook delayed type hypersensitivity response at 48 hours. Now, how do we study this? Then we're doing, from this biopsy, we're doing an RNA-seq. We have some preliminary information, but I'm not going to show it because it's not very informative right now. The only thing that I can say is that the principal component analysis clearly showed that there is a significant difference between all the samples from all insects that are at 48 hours when compared to 4 hours. Then there's a signature of molecules that are upregulated in the skin of humans after 48 hours that seems to be very common in different insects, but we need to figure out what are these molecules. We're still, this data is very new, we're still analyzing that. But also we wanted to know what type of cells are present there, or what type of cytokines or cells markers that in the skin of these individuals after an insect bites. For this, we use a new technology that is called digital spatial profiling from NanoString. And why we decided to go with this technique right now, although we're using other techniques of high throughput multiplex histology analysis too. But in the meantime, we're using this technique because it allows actually to use parafilm samples. You can get the parafilm samples, send it, stain it with these markers, and then run it in the instrument. And the instrument is going to recognize, select what is called region of interest. Oh, sorry, there was a title here, but it's not. This is the unbeaten skin site, and this is the beaten site. Okay, I'm just gonna show you. This one is this side, and this one is this side. Okay, just to give us a reference. And it's not exactly the same slide, but it's the same proportion of cells. Then clearly here we start seeing a lot of cells, T cells probably coming, and that we didn't see here. But we selected what is called the region of interest, these two, and asked the question, what's the difference there? This approach is quite interesting because it is a mix of markers for that is used for RNA, but also is proteins that are binding to the to the molecules in the skin, antibodies that are binding, and then the signal is through these fluorescent groups in the antibody. Then what you read is actually this. Then if we compare the unbeaten with the 48 hours, we can start seeing already something very different. We see this high expression of CD44, 45, stone, sting is a very important element we want to show. And this is just one example. We have too many to go. But what we're seeing now is actually the first type of study in humans that will allow us to understand what happens after an insect has beaten us, what's happening in the environment that is where the parasite, the pathogen is going to be, and more important, what happens to the environment for a vaccine. This is what the vaccine is going to face, this environment. Now, what can we do about it? And that's what we want to try in the future. Then just to close, I hope that at the end it convince you, Sharon and I, and also Hira help us a lot with many of, and Renadir, of the importance of the infection inoculum and our efforts, not only to understand this, but also how are we translated this information in human clinical trials to allow us to get better vaccines, to get some vaccines, a combination of vaccines. I just want to acknowledge a lot of people. I have a very active and fun lab, and I'm very fortunate to collaborate with two amazing scientists, Sharon and Fabiano. They're always we're always brainstorming and directing different projects and sharing information. Of course, all the group at the FDA, Renadir and Hira and all the group, the clinical study, Matt Memoli, and Jessica now is part of the LMBR. She was here, but now she's actually in Cambodia. And the viral immunity and pathogenesis unit help us to do all the in vivo imaging for the neutrophil, all work in Brazil that I didn't show actually, and many other people that has helped us a lot in these different studies. And with this, I'll thank you again for your attention, and I'll be happy to take the question.
Thank you. Thank you very much, sir. It's an excellent lecture, and you have shared your experience for the development of vaccine development, the universal mosquito development, which is utmost necessary for the countries like tropical countries like India. And now I would like to request Dr. Somaditya De, who received his PhD from West Bengal State University, and now he is an assistant professor in an undergraduate affiliated undergraduate college, Barasat Government College. Somaditya, please take the charge.
Thank you, sir. Thank you. Thanks a lot, sir, for such an interesting presentation. With your permission, I would like to start the interactive session.
So many questions.
So there are several questions. The first question is that priming with a salivary protein only or boosting with both the salivary antigen and Leishmania antigen will better drive a Leishmania specific immunity?
The question was if combination of a Leishmania antigen with a salivary antigen will be better vaccine. The answer is actually yes. Okay. We already tried with Sima Rafati in Iran, and we showed that the combination of Leishmania tarantulae antigen with Senfly antigen produce a better immune response. We have a paper actually with Hira showing that combination of LGM19, the protein with the central knockout, actually was very strong and better than any of the two alone. Those were done with needle, but we need to show with Senfly bites. But what we're seeing, and that's probably what we need to emphasize, I don't think we're going to see, or I'm envisioning a vaccine, salivary vaccine working alone. I think that it's going to be different components, the pathogen component and the saliva component, yes.
Okay, so thank you. The next question is that, sir, have you identified any protein in the saliva of sandflies which have a lytic activity on the parasites, like those analogs to the lysozymes in vertebrate saliva?
No, that's a very good question. We found molecules that bind parasites, actually. We found molecules that help the parasite through neutrophil inhibition of the neutrophil traps, but I have not detected any salivary protein that can kill or affect leishmania. In fact, I don't think that will happen because we already tried adding leishmania saliva, all the molecules to leishmania, and we didn't see any effect in killing.
Okay. Okay, sir. So there are now two related questions. What are the effects of the salivary components on the regulatory T cell subsets as well as the Th17 subsets in vaccinated mice?
I don't remember in mice we saw that. We saw a Th17 upregulation in the monkeys, in rhesus macaques. Regulatory T cells, I don't remember much about that in particular. I think we need to study. I mean, the problem with the monkeys was very difficult to get all the reagents. I think what we're doing right now is actually, that's a very good question, because we are going back into the mouse model to ask these questions because there are more tools. But I think those are very valid questions, and we just needed a molecule that it consistently protects, and that's what we need. And that's what we are doing right now, at least for the mouse model.
Okay, sir. Okay, thank you. The next question is that whether is it possible to characterize the duration of protective memory generated by the vaccination strategy with any particular recombinant salivary antigen?
That's a very good question. We did that. We did that a few years ago with the salivary proteins, and we wanted to know if this was a long-lasting protection. Then what we did was we immunized the animals with the salivary protein only, like, I think it was like 100 nanograms or 1 microgram. It was very little, and waited 5 months. 5 months, and then challenged them. Then we did 1 month, and then we did 5 months. And still, we could see not only we saw the correlates of protection, active CD4 T cells, interferon gamma, but we saw a very high level of protection. Then, yes, we have done the long term, and imagine in the mouse model, 5 months is a long time. Then we can do the same with other models, but yes, the answer is yes.
Okay. So the next question, is there still any study of vaccination with Phlebotomas argentipes salivary proteins followed by the challenge with Leishmania donovani infection?
Yes, there is already published, and we are collaborating with a group in India actually to do this. We identify actually a fellow from India, Samiran Sa, went to the lab and helped us to identify salivary proteins that are inducing a Th1 response. Then we are also collaborating with other groups. We are finishing some experiments also. The problem with argentipes in our lab is we don't have a colony. We don't have a large colony where we can do this. Then everything is being done with needle. And we're trying to see if we can increase. Sharon has a very nice project in India right now, in Bihar. And one of the things she's trying to do is to see if we can collect the sandflies there and create a large colony there and do other experiments with infected sandflies. Then it's been challenging because for argentipes colony is not that easy to have. But the idea, yes. And the paper that show argentipes protection or the correlates of protection is actually, they showed that it was the same thing we identified, SP15. What we found in the SP15 family of proteins in sandflies for cutaneous leishmaniasis in monkeys and actually in humans in Mali, the group observed also for Phlebotomas argentipes. Then these proteins may have something very common in the structure.
Okay, sir. Okay. The next question is that would you be looking at the monocyte macrophage system at a later stage as an effect of the vaccination strategy?
I think we're going to see everything related to the immunity and active monocytes. Yes, we have to see those. That's what we needed to do with the mouse model. We needed to understand better the initial events and the late events. Definitely so. We just needed to have a right molecule.
Okay, sir. So another question, how many bites necessary for infection occurs in human in case of sandfly or in case of mosquito? So,
Oh, one. One bite should be more than enough.
okay, sir. Oh, it's okay. Another one, sir. Another question, sir. In vector-infested areas, do natural immunization occur? There should be more exposures to bites and hence vector saliva in vector-infested area. So I think the
That's
question
a
is,
very,
yeah.
yeah, no, I understand the question. That's a very good question. We always all brought are asked that question. I mean, if people are being bitten, will you expect to see less disease? And what we observed in Mali actually was when we screened all the individuals that were bitten by sandflies, we noticed three categories. We saw people that, by PBMC, not by skin. Remember that when we did the skin, everybody that produced the TH had this TH1. However, if you take the blood of these individuals and you stimulate with salivary proteins, we noticed that 30% has TH1 response, other 30% has TH2, and then another 30 something percent has a mixed response. Then we hypothesized that probably 30% may be protected. And I think that's a number that you probably see. I mean, there's a good correlation. We don't have any proof of that. But what I'm trying to say here is that not all the individuals are going to respond equally to the bites because it's a mix. Then what we hope or what we hypothesize is that if we immunize these individuals with a specific salivary proteins, that will prime the immune system to have all the time TH1, that when the insect bites again, it's going to be into the TH1 concept. That's what we're looking for. Then the bottom line is not all the individuals are going to respond the same. Our goal is to make them to respond the same.
Okay, sir. The next question is that what is the role of eosinophils in special leishmaniasis, especially in the case of delayed type hypersensitivity development?
In eosinophils do not participate in the delay type. I think they're more into the other responses. And to tell you the truth, I do not know what will be the role of eosinophils in leishmaniasis. I don't know that. I'm to be honest here. But what I know is that we're looking for responses that are very clean, that are CD4, that do not have eosinophils, because you don't want to have eosinophils and other like mast cells that will produce TH1 or allergic responses. We're trying to avoid those cells. Then in most of the studies, in the monkey studies or other studies we have done, we have shown actually that they do not produce, or they didn't bring, with the specific salivary proteins, they do not bring eosinophils or other cells that could produce this allergic response. It's mostly T cells and macrophages.
Okay, sir. And there is another question, sir. How do you compare the natural infection through sandfly bite versus using a fixed number of parasites against macrophages?
How do I compare in terms of infection, maybe?
I guess so, sir, in terms of infection, right?
Well, I think Charan was very clear, and I mean, we did the same. When Charan, I mean, in the experiment that Charan did, and what really showed that when you have only the parasites alone, there's no visualization. There's no the parasite is alone waiting for a neutrophil to come and help them. When you have the bite, you start having not only the saliva, the microbiota, the neutrophil factor that will bring neutrophils and help. Then there is a significant difference. There is a significant difference in the way that the antigen is going to be taken up, in the way that the disease is going to initiate. Yes, I mean, there is definitely needle versus bite. Yes.
Okay, sir. Okay. Thank you, sir. Thanks again for sharing your vision with us. Much of our doubts are clear now. So I would like to end this interactive session here and would hand over to Dr. Chiranjib Pal sir for continuing the rest of the program. Thank you, sir. Thank you.
Thank you, sir, once again. And I have a question just to ask you. In India, both Phlebotomus papatassi and Phlebotomus argyropus, they coexist in the nature. And papatassi, they are anthropophilic, but Argentipes, they are zoophilic. So the probabilities of getting bitten by Phlebotomus papataceae may be higher in reality. So do you find, is there any role of papataceae bite on transmission of Argentipes mediated Leishmania donovani infection?
That's a great question.
I have asked you on the last day
the,
during
the thing
2007.
is this. The thing is, we don't know. We don't know if these proteins cross-react. We have some evidence of other proteins cross-reacting. For example, we were very surprised that Phlebotomus perniciosus in Europe cross-react with some of the proteins of Dutomya longipalpus from the America. It was at the cellular level. It was incredible. Then I think it would be very good to test if Phlebotomus papataceae and Phlebotomus duboski cross-react. We know that Phlebotomus duboski and papataceae cross-react, meaning that people that are bitten by duboski are going to be protected against papataceae. But I do not have any evidence if an immunity produced or anything produced by papataceae will have an influence, at least for protection, to Phlebotomus argenteus transmission. I have no idea, but if there is some similarities and cross-reactivity, they should protect. But I don't have any evidence for that. But that actually is something that needs to be done.
Yeah.
Actually, that's a very good point. That's a very good question.
We're trying to develop this experiment. Let's see when the lockdown will be over and the COVID crisis will be over. Then maybe we can go through these experiments.
Yeah, I think that's a very valid question, particularly because if we know that the SP15 type family is protecting or is showing some level of protection for Donovani, and if the SP15 from papataceae and Argentipes are very similar and they may cross-react, I think there's a potential of cross-protection. It needs to be tested. But as I said, I mean, Peter Wolf already showed that animals that were exposed to Duboski bites or glands, they protect against papataceae major glands. I mean, it was very clear. Then, and we know actually there is cross-reactivity in the SP15 molecules of Duboski and Phlebotomus papataceae. But the Argentipes is a very good question. I like it.
Yeah, thank you, sir. So another question: there are not too many work on Sargentomia. However, they are not not anthropophilic. They may prefer the reptilians. So, with your experience, do you find that Sargentomia salivary gland components or the microbiota, they may also have some role in either transmitting or disseminating the disease?
That's a good. I mean, there's some Sargentomia, at least when we went to Mali, and that was one of the most dominant sand flies, the Sargentomia, and particularly Sargentomia swetzi. And those sand flies are anthropophilic, actually. Then, now, we wanted to test to see if actually, but we never had a chance to do that. But that's a very question, too. I mean, will a sand fly species that is very abundant, like Sargentomia, will it have any impact on affecting the disease progression or transmission of other sand flies? That's a very good question. I remember we wanted to address that, but we could never do that. I think the person that is involved is studying Sargentomia, I think is Peter Wolf and Paul Bates. I think they decided to move to do more of the transcriptome of the Sargentomia to find what molecules, and apparently they're very similar to I don't know to what level or how many, but they're very similar to the other sand flies. Then it will not be surprising that it could be some cross-reactivity because the molecules that I show in papataceae or Dutomya, they are also found in Sargentomia.
Thank you, sir. So now I would like to move on to the vote of thanks. I would like to say Dr. Hira Nakashi discussed the way of developing the second generation vaccination against the deadly parasitic disease leishmaniasis. Dr. Kamahi discussed the complex interactions between vector-derived factors and the host immune system during the sandfly bite. Dr. Valenzuela discussed the basic and translation research of vector salivary proteins leading to vaccine development, the concept bench to bedside. Being the scientific community of tropical country, the vector molecular biology need to be explored more for the therapeutic inventions. With this hope, we would like to announce the ending of this session and beginning of our more concentration on vector molecular biology in our country. Thanks for your interest and joining in this meeting. Thank you very much, sir. Thank you, sir.
Thank you very much.
Thank you, sir. Thanks for joining. Thanks for joining. We are privileged to
Thank
have
you.
the experts like you. Thanks for joining.